But by the request of my colleague, Vice Chancellor Pethani, I am with you. I congratulate Saurashtra University and the people who have joined with this webinar, sponsored by GUJCOST, role of multi-component system in pharmaceutical formulation department on development. This is a time before pandemic period, corona, during the time of pandemic and corona. This is a high time to think and to research how to face this critical time, because it is a long period, 10 months. We are trying to research the solution of pandemic, a medicine, a vaccine. In all universities, we have a pharmacy department. We are lucky to have very big institutions like GUJCOST and all others, better than the institution of world even. As Pankaj Bhai from GUJCOST is trying to research the medicine for this pandemic corona. By this webinar, I think you all are going to present your papers, solutions, what you have done, what your professors, what your students have done in this regard. Again, I congratulate the sponsor agency GUJCOST, the participant GTU, Saurashtra University, and other universities who have taken part. I wish success of the webinar. This is the second day of 2021. So I pray God for success in our work. Good day. Thank you.
Thank you, sir. We are very lucky and fortunate that you are with us. Your blessings and presence will means a lot for us. Now, it is a matter of great pleasure and pride to welcome chief guest of our today's function, Professor Dr. Naveen Sheth, sir, Honorable Vice Chancellor of Gujarat Technological University and founder head of Department of Pharmaceutical Sciences and ex-dean of Pharmacy and ex-member of GPSC. Sir is the Vice Chancellor of Gujarat Technological University from December 2016. A renowned administrator and educationist, Sir started his career way back in 1979. During the course of time, he emerged as a tireless academician and herbal scientist. Sir has obtained his B.Pharm and M.Pharm degree from LM College of Pharmacy and PhD degree from Saurashtra University. Sir has also served as a member and chairman of various committees of UGC, AICT, TST, GUJCOST, and several universities in India. Sir has been the advisor and later on the member of GPSC. Sir has attended many international and national conferences in India as well as abroad. His research interests include traditional drug, herbal drug, and standardization. Sir has more than 100 publications, including 65 technical papers in renowned research journals, 42 papers in national conference, and 21 papers in international conference. Till date, 28 research scholars have successfully completed their doctoral research under his guidance. At present, 4 research scholars are pursuing their PhD under his supervision. Till date, Sir has been awarded with various prestigious awards like Vice Chancellor of the Year 2020 by Integrated Chamber of Commerce Industry, New Delhi. Sir is also awarded with a Lifetime Achievement Award by KLE University, Karnataka and World Education Convention 2017. Sir is also awarded with Swami Vivekanand Excellence Award and Academic Excellence Award. Now, with this brief introduction, I would like to request Sir for his inspirational word. Over to you, Sir.
Namaskar. I'm audible?
Yes, sir. Sir, you are
Yeah.
audible.
Namaskar. It's a very good morning, very good afternoon, good night to the all, because the experts and the participants, they are from all over the world joined here in this international webinar. So respected our Honorable Minister Shri Bhupendra Singh Ji Chudasama Ji, our Vice Chancellor, my colleagues, Sri Vathani Sir, Pro Vice Chancellor, Dr. Deshani Sir, Registrar Dr. Jatin Sir, the today's guest and CCVS Subramaniam Ji, the Head of Department, Mihir Bhai, all faculties, and all those who join virtually in this conference. So very first, I congratulate the team of Pharmacy Department, Saurashtra University and the organizer for arranging this wonderful webinar in this pandemic COVID-19 situation. And we see from the screen that 238 they are joining this Zoom directly. We can see on the screen, and there are number of participants virtually joined on Facebook Live. So it's a very successful conference. Second thing, the topic they have selected, that is also very, very important, because the general topics are selected by the most of the webinar organizers. But here a very, very typical topic is selected, and very important topic is selected as the multi-component. So that part is very important. To make a tablet, capsules and other formulations is very easy nowadays. But either the bioavailability of that formulation is good, either the compressibility is good. So many other factors, the solubility and other things, they are very, very important component and playing the role. So, and very few experts are available. So Mihir Bhai has really nicely aligned all experts from India and globally, particularly of these subjects. So definitely those youngsters, young budding pharmacists join in this webinar and faculties. It will be a very, very useful webinar for them. It will enhance the knowledge on multi-component solid form. It will also helpful in their research for the improvement of solubility, compressibility, and bioavailability. So I again congratulate the whole team and the Saurashtra University for arranging this webinar. Thank you very much.
Thank you, sir. We are really grateful for the time and effort. You took to share your thoughts and experience with us. Thank you, sir. Now I would like to welcome our Honorable Vice Chancellor, Dr. Nitin Bhai Pithani, sir, for his inspirational word. Sir, over to you,
नमस्कार
sir.
फ्रेंड्स, 2021 की साल मेरे सबके लिए नया किरण लेके आई है। आज सौराष्ट्र विश्वविद्यालय राजकोट के लिए भी गौरवपूर्ण दिन है। सौराष्ट्र यूनिवर्सिटी के डिपार्टमेंट ऑफ फार्मास्यूटिकल के एक दिन का इंटरनेशनल वेबिनार की जो योजना बनी है, उनका इनॉग्रेशन सेशन शुरू हो रहा है। इस सेशन में हमारे बीच उपस्थिति है हमारा स्टेट का शिक्षा मंत्री सम्माननीय भूपेंद्र सिंह जी चुड़ासमा साहब, हमारी यूनिवर्सिटी के प्रो वाइस चांसलर डॉक्टर देसानी जी, फार्मेसी डिपार्टमेंट के हेड ऑफ डिपार्टमेंट यानी डीन डॉक्टर मिहिर रावल जी, ये पूरी कॉन्फ्रेंस का जो कोऑर्डिनेशन किया है हिरल मनानी जी, और आज हमारे बीच हमारे छात्र तो जुड़े हुए हैं। छात्रों को मार्गदर्शन करने के लिए मिनेसोटा यूनिवर्सिटी से आज हमारे बीच उपस्थित है डॉक्टर केल्विन सर। इसके साथ मेरे साथ जुड़े हुए हैं देश के जाने माने और साइंटिस्ट और हमारा एक्सपर्ट्स। दो बात कहना चाहता हूं। आज हमारी जो स्थिति है कोविड-19 की वजह से काफी सारी जगह जगह से हम टीकाकरण के लिए प्रयास कर रहे हैं। हमारी ये जीवन में काफी सारी मुश्किलें रहती है फिजिकली फिटनेस के बारे में और हमारी तंदुरुस्ती के बारे में। तो हम हर रोज अलग अलग प्रकार की मेडिसिन लेने के लिए प्रयास करते रहते हैं। अच्छी मेडिसिन मिले और हमारा स्वास्थ्य अच्छा रहे, वैसा काफी प्रयास रहते हैं। आज मुझे गौरव है और गर्व भी है कि हमारे विश्वविद्यालय का स्टूडेंट्स और हमारी फैकल्टी ने काफी सारा रिसर्च किया है और ये रिसर्च के वजह से सोसाइटी को काफी सारा फायदा भी मिल रहा है। मैं जानता हूं ये मेरा फार्मेसी डिपार्टमेंट ने डायबिटीज के लिए और काफी सारी जो हमारी मुश्किलियां है, समस्या है, उनके लिए दवाई और काफी सारा रिसर्च करके समाज के सामने एक अच्छा जो कंक्लूजन दिया है। आज मैं बताना चाहता हूं हमारी फैकल्टी या स्टूडेंट्स के लिए एक बड़ा मौका है। रिसर्च एंड डेवलपमेंट दोनों की दिशा सही रहती है। जिस डिपार्टमेंट ने या तो जिस विश्वविद्यालय ने ज्यादा रिसर्च किया और इनोवेशन किया, उनका डेवलपमेंट भी बहुत रहता है। फार्मेसी डिपार्टमेंट ने भी अच्छा रिसर्च किया। आज रिसर्च स्कॉलर के लिए और फैकल्टी के लिए उनका ज्ञान का ज्यादा बढ़ावा देने के लिए ए डिपार्टमेंट ने इंटरनेशनल कॉन्फ्रेंस का एक योजना बनाई है। आज ए इनॉग्रेशन फंक्शन पे देश विदेश से आने वाले एक कॉन्फ्रेंस में हमारा फैकल्टी यानी स्टूडेंट या तो एक्सपर्ट को मैं हमारे विश्वविद्यालय की तरफ से हृदय से स्वागत करता हूं। और हमारा जो एम्स है, हमारा जो लक्ष्य है, अच्छी तरह से लक्ष्य में प्रति हम आगे निकलेंगे। इस प्रकार का आपका मार्गदर्शन मिलेगा तो ये सोसाइटी के लिए, नेशन के लिए और हमारे जो विश्व के लिए बहुत उपयोगी रहेगा। मैं इस कॉन्फ्रेंस की शुभकामनाएं चाहता हूं और मैं धन्यवाद और अभिनंदन करता हूं पूरे डिपार्टमेंट ऑफ फार्मास्यूटिकल साइंस का फैकल्टी, कन्वीनर, कोऑर्डिनेटर या स्टूडेंट को फिर से मैं आप सबका हृदय से स्वागत करते हुए धन्यवाद की मेरी ये फीलिंग आपके सामने रखता हूं। जय हिंद।
Thank you, sir. Thank you very much for your motivational and encouraging word. Your encouragement will meant a lot for us. Now, with this word, we ended our inaugural function, and within a couple of minutes, we're going to start our technical session. Now, before we proceed for our technical session, I would like to convey one message to all the delegates that after each technical sessions, we have arranged a short question answer sessions. So you can raise your queries in a chat box. Thank you. Now, in our first technical session, we have with us Dr. Kelvinson, Professor of Pharmaceutics and Director of Graduate Study and Department of Pharmaceutics, University of Minnesota. Dr. Sun's research focus on formulation development of tablet products to appropriate material science and engineering principle, which includes crystal and particle engineering for superior flow and compaction properties, solid state chemistry and engineering, pharmaceutical unit operations. Sir currently served on the editor advisor board for Journal of Pharmaceutical Sciences, International Journal of Pharmaceutics, Molecular Pharmaceutics, and Pharmaceutical Research. He has served on expert committee in physical analysis of United States Pharmacopoeia since 2010. Dr. Sun is a APS Fellow and a Fellow of a Royal Society of Chemistry. He has received a number of prestigious awards, which includes Raf Sangrao Memorial Award by International Pharmaceutical Excipients Council, which is given for his outstanding research contribution in the study of excipients or excipients-related technology over a number of years. With this brief introduction, I would like to invite Sir to deliver his lecture on preparation and application of pharmaceutical co-crystal. Over to you, sir.
Thank you, Dr. Claudia. And I want to thank the organizing committee for the invitation. It's indeed a pleasure for me to be part of this great event. Particularly, I want to thank Dr. Ravo for the friendship and for the collaboration in the past, a few things. So let me try to share my screen. Can you see my PowerPoint?
Yes, sir, it is visible.
Okay, great. In that case, I will start. Okay, great. So thank you for the very nice introduction. I really appreciate that. So in my talk today, I'd like to first give a brief introduction about the relevance and significance of multi-component solids, particularly co-crystals. Then I want to spend some time talking about potential pharmaceutical applications of co-crystals. Here I mentioned the potential. You will become clear later on when I finish my presentation. And also then I'll switch gear to talk about strategies which we can use to prepare new co-crystals. Then finally draw some brief conclusions. So let's think about this. Earlier, it was already mentioned that the multi-component crystals play a very important role in drug development. So why is the case? One reason is when we develop a drug, the drug must have must meet quite a few requirements for certain properties. For example, mechanical properties, stability, and so on. But this optimal properties, the space of that is quite small. So oftentimes, if you get a drug candidate is in this space, it's a luck, it's a blessing. But many times, the drug solid you have in hand maybe have some property are optimal, but some other properties are not. But if it's not optimal, it's okay if the properties are acceptable. So you consider the space a little bigger, or include these less optimal but still manageable properties. But unfortunately, a lot of times you have drugs with one or more properties falls even outside this acceptable space. Once you enter this unacceptable property space, you run into trouble with making your product reliably and providing high quality. So therefore, if in those cases, with the red arrows indicating drug molecules with properties not acceptable. In that case, what we can do is ideally we take the drug molecule, we look at the molecule using solid state chemistry or crystal engineer. We try to engineer this property out to drive them into this optimal space like this. So this idea, but unfortunately, our knowledge is not there yet. So for now, if you go to talk with people in industry, a primary tool they have is really try to do the solid form screening. Early on in development, try to discover as many solid forms as possible. For that matter, the multi-component solids plays a very important role. Out of these different kinds of multi-component solid forms, co-crystals are important class of those. So here's my view of how these different materials fit together under this umbrella of multi-component solids. Because I should co-crystal first because that's the focus of today's talk. So this suggests a space for the co-crystal. Now you have hydrates. So when you have the water crystallized in the lattice and it's hydrate, but then the overlapping area is the hydrate of co-crystal. It's in the region 1. Now you can also that's the region salts. So the salt can overlap with the hydrate and co-crystal. Then the region between the co-crystal and salt is known as ionic co-crystal, or sometimes called salt co-crystal. Then the overlapping area between hydrate and salt is a hydrate of salt. And then very interesting, the region 4, so it's a hydrate of ionic co-crystal. So the region the space becomes smaller and smaller, and getting the solids become more and more complex. You have more components in there. And each of these, the 1, 2, 3, 4, and all these 3 classes, they each can have their own polymorphs. So in terms of chemical composition, is identical, but they can form crystals in different structure, form polymorphs. You can see the very complex landscape we're dealing with. So I want to talk a little bit about this so-called cap co-crystal. So this one is a part of these ionic co-crystals. Cap stands for the conjugated acid and base. For this, we know from chemistry, general chemistry, when 2 molecules differ by 1 proton, either molecule takes on 1 proton or delete a proton, the 2 molecules becomes a conjugated pair. And this can actually interact strongly to form a pair of molecules, which forms the building block of crystals. And if they do, those crystals are termed cap co-crystals. So you can think about you have salts, traditional salts, acid, base react to form salts, and traditional co-crystals, everything is neutral, then nothing is ionic. And when you combine these 2, like what I showed earlier in the Venn diagram, and this that formed the class of ionic co-crystals. Means in the crystal you have neutral molecules, you also have the ions. I mentioned the cap co-crystal, and everything else other than cap co-crystal in this ionic co-crystal space is just other ionic co-crystals. They can be prepared in different ways. So why do we care about forming co-crystals in the context of pharmaceutical development. There's quite a few things. Tabletability is a long list: stability, solubility, and dissolution, high hygroscopicity, stickiness to punch, and taste, and melting point, permeability, flowability, and many more properties. Really, the challenges are different for each compound. So every drug molecule is like a child. They all have their own personality, their own challenges. So let's look at some examples. So in terms of the tabletability, I don't know why it is okay. Okay, thank you. So so this shows a tableted plots of 3 materials. So this green one is that of. So why I see this drawing here? Okay, we need to have this under control. Okay.
Sorry to interrupt, sir. I would like request the organizer to have a control of screen, please.
Yes,
Thank
sir.
you. Yeah, it's okay. Thank you. So the green one is the tabletability profile of methyl gallate. So this graph, when you read this is the x-axis is the compaction pressure. You apply the pressure to a powder, you hope to form a tablet. And this y-axis is the strength of the tablet you form. So typically, you want the tablet to be high, has a high strength. So, but unfortunately for the methyl gallate, you can see even you apply relatively high pressure, the strength is not very high. So this means this material exhibits poor tabletability. But then if you look at caffeine, is another molecule, you can see it's better. But when you go above this 200 megapascal, you see the drop in tensile strength. So this one at high pressure can be problematic. But now if you form co-crystal between the 2 molecules, so this is the tabletability profile of the co-crystal between methyl gallate and caffeine. It's much improved. With increasing pressure, the strength increases. So this is what we like to see for a drug molecule, a drug crystal. So this just shows by forming co-crystal, you can improve tabletability of your drug. The reason for that is for the methyl gallate, which is exhibit very poor tabletability. The molecules interact with the neighboring molecules through hydrogen bonds. It forms a rigid 3-dimensional hydrogen bond network. Therefore, crystal is hard. It doesn't deform easily. That's why it doesn't form tablets easily. Versus the co-crystal, these molecules are packed into layered. This is rigid. Flat layer molecules are interacting through hydrogen bonds, so it's rigid. And because it's also flat with large separation between them, the layers can slide relatively easily. Therefore, this crystal is soft, it's plastic. So when you compress it, they can deform, they can bond, and therefore form a strong tablet. So the reason which we want to understand the mechanism or origin of the different properties is very important. It will come later. So same thing can happen to other drugs. Resveratrol is another important compound with pharmaceutical applications. You can see the resveratrol has a very poor tabletability, and if it forms a co-crystal with the 4-amino-benzamide, the co-crystal exhibits much improved tabletability. You can find many examples. We have done quite a lot of work on that. So other properties, solid state stability. For example, if your drug undergoes hydration easily, you start with unhydrated, but if you expose to high humidity, the crystal form converts to a hydrate. Given here is one example of caffeine. So here shows 3 curves. So this is a moisture absorption curve of caffeine. So this is a relative humidity from 50 to 95%. And this is a moisture content, amount of moisture gained by the sample at different RH. You can see the curves are separated, depends on temperature. At lower temperature, this hydrate can form more easily at the lower RH. But if you consider 25 degrees C, which is relevant to manufacturing, so this material undergo hydration starting from about 81% humidity, relative humidity. And with a higher RH, this more water will be absorbed and forms eventually, with increasing RH, it forms a pure hydrate. And this hydrate is a 0.8 hydrate. It's not a monohydrate. But this is the problem. This is problematic during granulation, water granulation. You put water in there. Some of these convert. Then during drying, maybe dehydration is incomplete. So batch-to-batch variation can cause problems. We like to get away from these kind of challenges. That can be done through co-crystallization. In this case, people co-crystallize this caffeine with oxalic acid. So this is oxalic acid. Through hydrogen bonds, they form the 2-to-1 co-crystal. By forming this co-crystal, they show, caffeine with high RH is not stable, undergo phase conversion within 1 day. But once you form the co-crystal, under some condition, it becomes stable against the humidity. Even 98% humidity still remain physically stable. So that's shows the advantage of this co-crystal. By forming co-crystal, you can also have the possibility to reduce hygroscopicity if your compound has such a problem. So this is one example from our study. This sodium benzoate is a salt. When you expose this crystal to more than 85% RH, and you start to pick up water very easily. This actually, the salt becomes a part of a solution, because of the deliquescence. But if you form co-crystal, it's resistant to high humidity, even at 95% RH. There's not much water uptake. And that shows the advantage of co-crystal. In terms of the, this is chemical stability. If you start with ammonium benzoate, it's a monosalt. When you try to study the moisture absorption property of this compound at 25 degrees C, kind of weird data were obtained. So with increasing humidity, typically, at higher humidity, you have more water get absorbed on the surface of the crystal or get incorporated into lattice. The weight should increase. But here, it actually decrease. So at higher humidity, something is being lost. It turns out this one, the salt undergoes degradation. So it forms, release the ammonia from the sample. Therefore, the weight decreases instead of increase. And once the ammonia leaves, actually what's remaining is the more stable co-crystal. This one is a capped co-crystal. Again, this is one example to suggest that a co-crystal can exhibit better physical stability over the salt. This shows the photo stability. So this drug is a natural furantoin. When you expose this one to UV light within this wavelength, you can see the drug degrade very quickly over time. But when you form the co-crystal, this degradation still occurs, but much more slowly. So that's an improvement. A little, you can see the clear stabilization effect by forming the co-crystal. And solubility is something people really care about a lot for drug delivery. So this one is just one set of crystals showing that. This red bar represents this drug, A. Solubility is measured here, given. When you form the co-crystal with different dicarboxylic acids, they give you different solubility. So this is A1. So this molecule forms a co-crystal with this acid, and so on and so forth. You can see that for these three co-crystals, solubility is much higher than that of the drug. So it has a potential advantage for drug delivery. Because of the nature of this molecule, you can see that this is drug molecule. It's a symmetrical molecule. This molecule is symmetrical. And also for the dicarboxylic acid is also symmetrical. So what happens is form the hydrogen bonded chains, molecular chains. When you change the length of the acid with different number of carbon, the basic structure doesn't change much, except this part just get longer or shorter. So but this change can still modify the property. You can see the solubility change from 0.5 gram per liter to very low. And when you have a more soluble co-crystal, they can potentially exhibit higher dissolution, faster dissolution, which is what we need for drug delivery. This one shows an example of carbamazepine. So this one is very poorly soluble. It forms hydrate. It doesn't easily dissolve. So this is a very slow dissolution rate. When you form the co-crystal with the nicotinamide, this co-crystal has higher solubility. But if you just put this co-crystal in water, the dissolution rate is about the same as the carbamazepine. What happened was it converts to the hydrate very quickly. So the co-crystal dissociate from the hydrate. Therefore, you lose the advantage. But if you do formulation properly, you can use some of the HPMCAs in this case, some of the precipitation inhibitor. You can prevent the phase conversion during dissolution. Therefore, you can take advantage of this higher solubility to get this faster dissolution. Punch sticking is another important phenomenon need to be avoided. This shows what the punches looks like after sticking occurs. You can see the punch, the metal surfaces are supposed to be shiny, smooth. You can make tablets over and over again, but in this case, after some tablets are made, material gets stuck on the punch. Therefore, you have to stop the process, clean the punch in order to make the new tablets. And these are what the tablets look like if you don't get rid of this punch sticking program. So definitely, this won't pass FDA's quality requirement, and patients won't take it. So this shows one example. Celecoxib is an anti-inflammatory drug, but unfortunately, this drug is a very high tendency to stick to the punch. This could be one reason. This celecoxib is marketed as a capsule, not a tablet. Maybe if you make tablets, you run into different issues. One of those may be the punch sticking. But the issue is now we know this drug has a high tendency to stick to the punch. What can we do with it? And as you can guess, we can just form a co-crystal. In this case, we can form a co-crystal with proline, and this co-crystal is much harder with different properties. You can see the much reduced propensity to stick to punch, and then you can see details in the references. So you can also consider making the crystal sweet by forming co-crystal. The basic idea is actually easy to understand. If you can form co-crystal with artificial sweeteners, then the crystal become sweet. We have shown this with salts. We make many sweet salts and also sweet co-crystals. It does help. So that's quite a few different kinds of sweetener you can consider. We have tried acesulfame and saccharin. These are the two most commonly used sweeteners. This shows some data with theophylline. Theophylline is is for the asthma drug. So, but if for pediatric patients, if you have they don't swallow tablets, they're very young. Typically, you make even a suspension, right? But unfortunately, the test is not good. In that case, in order to deliver drug to kids, sweet co-crystal becomes very handy, very useful. You can form a co-crystal with saccharin. You can also form a co-crystal with acesulfame. They both have sweet taste, so therefore you can give it to kids to form improved pediatric formulations. I want to point out here, you can see I have two arrows, one arrow pointing down, one arrow pointing up. This suggests the dissolution rate of one co-crystal is lower than theophylline, and the other co-crystal is higher. So I want to bring a very important point for you to consider. Co-crystals, we know at this point, when you have form a co-crystal, the properties will be likely different from the parent compound. But the changes may not always be desired. So the property either be enhanced or deteriorated. Why is that? The reason is that we need to think about these are properties we're considering, but what determines the properties? This can be thought of using this framework. I call this material science tetrahedron. It gives the idea about how to develop drug products and a lot of to guide research. But here, I want to focus on two things. You can see this tetrahedron have four tips. One is structure, one is property. What it says is the property can be many things that we mentioned earlier, and the structure can be molecular structure and different, depending on what you're talking about, can be different structures. But here they say crystal structure. So the crystal properties are determined by the crystal structure. So what we do is by changing the structure, you can change the property, and this property can in turn affect the performance of your drug, your product, and then you can also by doing engineering to modify the structure to attain the desired performance. So that's the essence of this material science tetrahedron. But what I want to mention here today is the property of co-crystals are determined by structure of the co-crystals. So co-crystal itself means not much. It really depends on what kind of structure you have. They can determine either the property can be either improved or deteriorated. So examples of deterioration in terms of tabletability. So this one is pyrosicam. It doesn't exhibit wonderful tabletability, but when we form co-crystal with saccharin, it's even worse. And of course, if you compare to saccharin, then the co-crystal exhibit much worse tabletability. And think about the solubility between the ratio of the solubility of the co-crystal to the solubility of the drug or ligand to the solubility of drug. So this is a log scale, right? That appears log linear. But what I want to point out is if you want to have an enhancement in solubility, you want your point to be above this line. So the ratio has to be more than 1. You can claim it has improved the solubility. To do that, you can see that means the conformer, solubility of the conformer in general needs to be at least 10 times that of the drug in order to see a improvement in the solubility of the co-crystal. Then when you consider the cases where you can form co-crystal with conformer with solubility not more than 10 times of the drug, is in this region. So the solubility ratio, solubility of the co-crystal to the drug is actually less than 1. So above this, the 10, you have an enhancement. Below it, you have a deterioration. Solubility goes down. But it doesn't mean it's bad because the properties are changed. You can just be creative. You can figure out how to take advantage of the lowered solubility. For example, you can make the drug release more slowly. Therefore, you can attain a sustained release of the drug. For example, this isoniazide is extremely highly soluble. Therefore, drug delivery is a problem because this drug also has a short half-life. Once you get absorbed, it gets eliminated very quickly. So you want to have the sustained release of the drug so you can maintain the plasma concentration over a long period of time. So then you can, fortunately, you can form a co-crystal with curcumin. We are familiar with curcumin. It's a very poorly soluble drug. It has a very low solubility. Now, if you form a co-crystal between the two, one is highly soluble, one is poorly soluble, you can get this solubility in between. You can, so it depends on the application. If you want to deliver isoniazide, you basically make the drug less soluble. Therefore, you can have a sustained release. So you can turn the disadvantage into a application. And think about the melting point. So this is a drug I mentioned earlier. And this one is the same set of dicarboxylic acids. when you look at the melting point of the coal crystals versus the melting point of these acids, you can see they follow out, very nicely follow almost a linear relationship. So the melting point of the pure drug A is about somewhere between 181 and 182. You can see that these 4 coal crystals exhibit melting point lower than the drug, and this one give you a higher melting point. So why is that? So now we can think about the binary phase diagram in the context of coal crystal. So here you have 2 molecules, A and B. They each have their own melting point. It's indicated here. And if these 2 can form coal crystal, then you have another phase in the middle. If it's 1-to-1 molar ratio, it's a mono coal crystal. You have a coal crystal here. So the coal crystal, and with each of these component can form a eutectic, right? You have this classic V-shaped curve. So this line is the eutectic melting temperature. So in this case, you can see the melting point of A is lower than the coal crystal, and which is lower than that of the B. So you have this rank order. But for when the phase diagram shows different features, you may have a different rank order. For example, in this case, the coal crystal exhibit high melting point. And so the melting point of A is less than B, is less than the coal crystal. And in this case, it will be the opposite. So the coal crystal has lower melting point than A and lower than B. So it depends on the phase diagram, the melting point can differ. So there's no reason to expect coal crystal must be higher, lower, or in between the 2 conformers. But I guess if someone does the statistical analysis, maybe more of the coal crystal will fall between, but we cannot expect that every time. So that's the first part. Basically, the 2 key points. Coal crystals can be used to solve pharmaceutical problems we encounter with drugs, because it does modify properties. Second point is, although properties can be modified, it doesn't mean it's always beneficial. So it can be better or worse than the parent compound, depends on application. So now I want to talk about how do we prepare it. Once you have a drug, you want to make coal crystal, how do you do it? I want to mention 2 things about in terms of crystallization. We make the coal crystal. You start with maybe 2 conformers. You want to form the coal crystal as a final product. It's about the crystallization. So we think about the crystallization in the energy landscape. So this is just qualitatively say all the different structures have different energy. And this free energy of different forms. So this one is a state. You start from a high energy state because it's not stable. If the energy is high, you tend to go to the lower energy state, free energy state. Say, for example, it can be a stable crystal. But on the way from this high energy state to the stable state, the path can be not smooth. It can be many metastable forms can be in between. So if you do the solution crystallization, you start with undersaturated solution, so that crystal will not be able to grow. So in order to grow a crystal, you can do a few things. One thing is commonly done is slow evaporation. When you do slow evaporation, the solvent evaporates, then concentration gradually increases. Then when your concentration goes above certain value, means the free energy of the system increase over time. Then when this one exceeds the critical value, you can form the crystal. So before the energy reaches enoughly high to cause the crystallization of these metastable forms, this stable form already crystallized. So this is one way to prepare a stable crystal, which can be coal crystal. But if you do this very quickly, so from the undersaturated solution, you bring the system by quickly removing the solvent, you can bring the system to the state with a very high degree of supersaturation. Then everything will start from here, very high energy. You can go to A, B, C, or they can find different paths to find different metastable forms. So let's talk about how to prepare the stable forms. Here we use the thermodynamics. So whenever possible, always try to make sure thermodynamics is on our side because it's predictable, it's reliable. In terms of the Koch crystal, on this graph, I showed that this line is the solubility line of the Koch crystal because it's the multi-component phase. So the concentration of one conformer depends on the concentration of the other. That's why the curve is down like this. And you can think about the KSP is constant, solubility product is constant. So in this case, you have solubility of the Koch crystal, and depends on where you are on this phase diagram. So this line indicates the solubility of the conformer A, means concentration cannot be much above this value because then it's not stable anymore. And this line indicates the solubility of conformer B. So this region indicates the region where the concentration of A and B in the solution is below the solubility of both A and B, but it's above solubility of the Koch crystal. Therefore, when you imagine when you suspend the solid A and B in your solvent, so the solvent will be saturated by A and B. So you have concentration here and there. So your system is in this state. So concentration of the A and B are here. And this one is above the solubility line of Koch crystal. Then this will, the free energy will drive it. This will gradually convert. You have starting with pure A and B, that will convert to the Koch crystal over time. So this is a very reliable way to prepare Koch crystal. You can, if you use the right amount in this experiment, you can get a fast pure Koch crystal. But sometimes, if you want to get a metastable Koch crystal, what can you do? Then you can use this trick of removing the solvent very quickly. So you artificially introduce the state of high degree of supersaturation and let it to gradually crystallize. Then you can form the metastable crystal form, as I mentioned earlier. So this one is in the laboratory is commonly available. The rotary evaporator. You can control these conditions. By doing this, you can really remove the solvent very quickly. The process is analogous to precipitation. You basically induce high degree of supersaturation and let the system crystallize out. But this one is more controlled. You can control temperature, vacuum, and solvent. And by using this, you can, as I mentioned earlier, you can hopefully, most likely, you can get this metastable form this way. You can prepare enough material for thorough characterization if you need it. So maybe I will skip this one. Okay, so by doing that, you can prepare a bulk amount of Koch crystal. So this Koch crystal may be metastable in solution, means that by normal evaporation, you cannot get a fast pure, or even you cannot even get Koch crystal. But once you prepare it, a Koch crystal in solid state is stable. So by doing this, for example, the rotoweb, you get a bulk quantity of Koch crystal. You can start to use that for formulation purpose. In this case, so ibuprofen and nicotinamide in solution is not stable, but by doing the rotoweb, we can prepare fast pure Koch crystal. You can study the property. This shows the tabletability of the Koch crystal is much better than both conformers. So the second method, right? First method is the thermodynamics. You get a fast pure stable form. Second method is using the fast solvent removal. You can get a metastable form. So third method is based on thermal property of solids. And I showed you the phase diagram before of a Koch crystal. But when you think about if you start with a physical mixture of A and B, and you try to heat it. So before the Koch crystal can form, right, you start with only A and B, so that these two will undergo eutectic melting first. and this occurs at a lower temperature, lower than both A and B. So once you have this, when you heat from room temperature up, then when you reach the eutectic melting point temperature, and liquid start to form. So the moment the liquid start to form, actually now, so the liquid is at a temperature below the melting point of the core crystal. That means the liquid is not stable actually, then you will crystallize into the core crystal. That's why you start with the A and B physical mixture, you heat it, once you pass this eutectic melting temperature, then you form a liquid will crystallize to form the core crystal. And this can be captured by DSC. So this is one example with salicylic acid and caffeine, and they form a 1-to-1 core crystal. And these are DSC thermograms that have different melting point. Now, when you start with a physical mixture at different ratios, 2-to-1, 1-to-1, 1-to-2, you can see in all the cases, all three cases, you can see the first endotherm. This indicates the eutectic melting is occurring. So it's this temperature. So once it occurs, then some of the liquids will also crystallize, but it's buried under this endotherm, because this endotherm is a sum of two events, eutectic melting and crystallization to form the core crystal. But once you pass this event, what happens is that the core crystal is here, right? And core crystal will melt. That's why you see the three peaks, these three samples all melt at same temperature, correspond to the melting point of the core crystal. Okay, so the DSC method. You can also do this with a hot stage microscopy method. And this really micro amount, it's even really microgram of material can be used to get this information. To do this, you prepare on the glass slide. You prepare a sample which consists of molten compound A and C with a zone mixed zone. And then, for example, you can melt them, heat them up, and you have a physical mixture, you heat them up, and they're both melted, and then you lower down temperature, they will solidify, and at this interface, so liquid will mix, form a zone of a mixture, and which is hopefully can become core crystal. And then what you do is you try to heat up this sample, and then because different phases have different melting point, you can see that what's happening on the microscope. So this is one example of ibuprofen, the S isomer, enantiomer, and this C is nicotinamide. As I mentioned earlier, they can form 1-to-1 core crystal. They have different melting point. So by preparing this sample, you can see the A is the ibuprofen, C is the nicotinamide, the B is the core crystal. So when you heat it up slowly, so at 47 degrees C, all three phases are present. They didn't melt. But when you raise the temperature to 51 degrees Celsius, you can see this phase start to disappear, and you have but the core crystal phase still remains. When you heat up to 85 degrees, even core crystal also melt. So only the nicotinamide crystal left. So from this phase behavior, it's okay. This phase is the core crystal phase because it has different melting point. Lastly, I want to talk about the cap core crystal. This is special because I mentioned the conjugated acid and base that only differ by one proton. So because of that, you can think about, say, if you have a acid-base reaction, the acid can transfer proton to B to form this conjugate acid and conjugate base of the acid. And in this case, HA and A minus will be considered a conjugated CAB, right? A conjugate acid and base. Or B and BH will be considered a CAB pair. The reason this is important is because between the two, it can form very strong hydrogen bonds because between them, there's a proton, right? So these atoms, these two molecules undergo in hydrogen interaction, which is charge assisted. So this normally give you very strong hydrogen bonds. That means that typically symmetrical, means the hydrogen or proton here will be in the middle of the two two atoms, and they have very short bound distance. And in fact, this one is known so far, is known the strongest hydrogen bond so far. So by taking advantage of this, you can imagine in solution or in liquid form, the CAB pair will always try to find each other because of high strength of interaction. So you can, for example, if with this molecule, with naproxen is the acid. Now, if you can try to crystallize this one with sodium salt of this acid at a 1:1 molar ratio, then we will find each other and crystallize out as a chemical crystal. Or if you start with the acid, you add a half equivalent of the sodium hydroxide to basically to deprotonate half of this molecule. So in the solution, you have still equal neutral and ionized species that will also crystallize out. At the end, it forms a 2:1 salt, or known as a halide salt. So, and their hydrogen bond is strong with a short distance, almost a linear relationship. Same thing for the base. I'll skip the details there. Same idea though. Okay, so that works well. But in order to do this, to satisfy this molecule being conjugated acid and base, there must be everything else the same except the different proton. But what we think was, okay, if we can keep this hydrogen bond symptom, but we can modify the functional group on the other part of the molecule. Therefore, we have this flexibility with chemistry. We can have the diverse chemistry and different molecules. We can still crystallize into a new crystal form. This is what we call the pseudo-cap co-crystals. For example, between the HUA, the acid, and the conjugated base, HUA and A minus, you can form a cap co-crystal. But now, if you on this acid molecule, you introduce a functional group R1, but on the other molecule, you introduce a functional group R2. So they are chemically different. They're not, strictly speaking, they're not conjugated base and acid and base, but this symptom still there because you have this carboxylate and carboxylic acid that still can interact through hydrogen bonds, strong hydrogen bonds. By doing that, you think about the conventional salts. This is a space you can operate within. It's a very small space. By doing the chemical crystal, you enlarge the space for the solid forms. By doing the pseudo-cap co-crystal, that's in theory, there's an infinite chance. Space can be very, very large. Just show use these three molecules that are structurally similar with this same carboxylic acid group there, but on this benzene ring, the different functional groups. You can form co-crystals among those, so that they still have these strong hydrogen bonds like here, but they have chemical diversity there. So it's a pseudo-cap. So that's the fourth method. I think about using the hydrogen bonds to really design new crystal forms. So to conclude, so crystallization it can be a very effective approach to solve a problem we see in formulation in delivering drugs. But one thing I want to mention is that not all co-crystals exhibit properties we want. So maybe it will, but because when you try to do it, you face a problem. So any change will likely to be better. So, but we should not expect that. Don't take it for granted. And when you try to prepare a stable fast pure co-crystal, consider doing a slurry experiment. You will try to do the suspend both conformers in a suitable solvent. That will, thermodynamics on your side will drive the reaction to completion. Over time, you get a fast pure material. And if you want to get a metastable co-crystal, consider very fast solvent removal because you're trying to introduce a high degree of supersaturation before the stable form has a chance to crystallize. It's already at a very high energy state. Then the metastable form will have a chance to come out. And using these thermal methods, either DSC or hot stage microscopy, I mentioned earlier, they're very useful technique. They don't use a lot of material. You can do a lot of screening, use a very little material. This is extremely useful for pharmaceutical companies at the early stage of development. That's when you don't have a lot of APIs available. The drug is very little material available for screening. Then think about by taking advantage of the strong hydrogen bonding between the conjugated acid and base pair. Now, this is a charge-assisted hydrogen bond. It's very strong. You can really design new materials. And this also, if the drug is acid or base, if you do this in hemi salt, the potency of the co-crystal will be greater than normal salt because it's only 2 drug molecule versus one kind of contact air to form the salt. So before I take questions, I'd like to thank people who did the work and my collaborators who contributed to the work I presented you today. And a lot of crystal structure, everything was done in the X-ray crystallography lab at Minnesota, and also the Minnesota Supercomputing Institute will have the resources accessible to us. I'd like to thank the various organizations who had funded our research in the past 20 years. I cannot believe it's a long time, no, 12 years. So, yeah. Thank you for your attention. I'd like to take any question you may have. Remember, this is just I'm just scratching the surface. In each of the slides I showed you, there's a paper behind it. You had to go there, read into it, think about it. So now I'm open to answer any question you may have.
Thank you, sir. Thank you very much. You had nicely explained the use of co-crystal to solve the pharmaceutical problems like stability, tabletability, dissolution, with example of each and every problems. And you had nicely explained the preparation of a stable co-crystal. So now we had several questions from the participants. Should I read that?
Yes, please. Yeah.
Now, the first question is asked by our speaker, Professor Dr. A.K. Bansal, sir, who is a professor and head of pharmacy department of NIPA. The first question that sir had asked is, for ionizable compound, sometimes both salt formation and co-crystals are possible. Which solid form out of salt and co-crystal is likely to provide higher solubility advantage?
I would say, yeah. By the way, I know Professor Bansal very well. We're very close friends also. Yeah, and so thank you for the question, Professor Bansal. Yeah. So I would say the both salts and co-crystals, they in terms of the range of their solubility can be very wide. Depends on the either the contact ion or the conformer you're using. But I would say in general, my own feelings, overall on average, the solubility of the salt probably will be higher than co-crystal. But remember, actually, even for salt, it really depends on the interaction strength, the strength of the crystal lattice. And there are salts with a very, very poor solubility. So they are insoluble salts. So, again, it's very broad for both class of materials. It's really hard to say. It has to be compound specific. Yeah. But if the things are very similar, because you have, for the pure salts, you have these all ions in there. So when they go to solution, they can interact with the water more strongly. That's, it's kind of favored to go to solution. But when we consider solubility, we need to consider the strength of the crystal lattice. So it's everything together. So there's no easy, clear-cut answer for that to that question.
Thank you, sir. Now, the second question is also asked by the sir. The area of sweet co-crystal is very interesting. Is sweet co-crystal sweeter than physical mixture of API and the sweet co-former?
That we didn't examine, so I don't know for sure. But the reason I think it's the advantageous to use to form either sweet salt or sweet co-crystal is you can deliver the sweetener at a molecular precision. So every drug molecule you have sweetener with it. People have in formulation, people use say for the chewable tablet. It has been done before. People use artificial sweetener as a part of formulation, but distribution is not very uniform. That means that it's possible you can have this, say, bitter drug is released, but without any sweetener. So, it won't be a very nice feeling for patients if you do a chewable tablet. But when you have this sweetener goes along with bitter drug molecule that dissolve at the same time, so therefore, in that sense, is more effective. So, if possible, then of course, by doing the co-crystal, you can also address some other potential issues, like what I showed with theophylline. With the two sweet co-crystals, you can, one has increased dissolution, one has a slower dissolution. So it depends on the application. You can choose one, not just the test, also some other properties.
Okay, sir, thank you. Now, the next question is asked by our delegate, that is Dhruv Patel. He asked that, what is the process for selection of a co-former?
Yeah, there are different ways to do that. Ideally, you look at the molecular structure. So that's some people have done. We have this Cambridge crystal structure database. That's millions of crystal structures. So people have done the data mining. They say, okay, this functional group and that functional group tend to form in crystal through hydrogen bonds and other interactions. So they call those symptoms. So almost like a formula can use, when they are present, they tend to interact with each other. Therefore, when you look at your drug molecule with certain functional group, you say, okay, and this functional group likely to form, crystals with some other functional groups. Therefore, you can select the conformer that way. So conformer with a certain functional group, which will tend to crystallize with your drug molecule. There's one way to do that, but it's still, for pharmaceutical purpose, the list of compound must fit in this so-called GRAS list, right? Had to be safe. So that's the number one requirement, and so that list is known. So you can choose from that list, and by combination of that and look at the chemistry, you can do. It depends on how much time you have, how much material you have. You can do a lot of screening that way. So that's how I would choose the conformers. Ideally, you choose a conformer based on chemistry with the hypothesis in mind. Then you try to test the hypothesis. That's how we do research. But sometimes it's difficult. Then you just do the brute force, just screening many, tens or hundreds if you want. Yeah.
Thank you, sir.
Sure.
Now, the next question that is asked by our faculty, Ms. Rajeshri Patel. The question is, what kind of thermal phase diagram produced by some unique co-crystal which have a lower melting point as compared to participating components?
That depends, right? I mean, I can find examples on that. I want to mention one example is there's a class of solvent known as a deep eutectic solvent. So that means the materials are solids, right? But when you mix them, they form a liquid. That means that the melting point, imagine they form co-crystal or salt, and their melting point is below room temperature, or it's low. So there are plenty of examples of the co-crystal or salts having melting point lower than both conformers.
Thank you, sir. Now the next question is asked by the Dux Jain. Co-crystal will increase the cost of medicines, and if not, then why the industries are not using it?
Yes. I think the philosophy is that if something works, why change it, right? if the drug molecule, just say neutral molecule, the parent molecule works fine. If everything falls in this acceptable range, as I mentioned earlier, then maybe co-crystal will make it better, but it's good enough. You just move on, right? People in the industry, they tend to, if they don't have to do something, they don't want to do it. Unlike academic, right? We want to really drive to the bottom of it. In industry, different. That's driven by the time, the resource, and a lot of other factors other than science. If they don't have to do it, they don't do it. But on the other hand, people are, nowadays people are more familiar with the concept. They start to do screening of co-crystal early on and do evaluation. And if it happens, the co-crystal exhibit much better overall property, it's not surprising if they choose co-crystal to develop. And there are more and more examples of co-crystal. Some are already marketed, So I think that this will pick up momentum. It's a matter of time, Eventually, you can just like a small molecule or macromolecules for therapy, right? Before, small molecule was dominating the market, but then at some point it's saturated. That's very typical to develop small molecule drugs. Then the protein and other therapies start coming to place. But eventually, reach equilibrium. You have all these coexisting. The same thing for these solid forms. I think the co-crystal salts, hydrates, neutral parent crystal, they will all be there. Really, case by case. Yeah, but I think the one hindrance before was in US, the FDA, they didn't have a very clear guidance on co-crystal. So basically, FDA treated co-crystal very badly. So it makes it very difficult. No very good, a lot of motivation for companies to pursue that. But later, the FDA clarified the guidance and basically cleared the path for developing a co-crystal. But that happened, not long ago. That's why everything takes time before we see, a wave of co-crystal being developed. It may take a few more years. Yeah.
Thank you, sir.
Sure.
Now the next question that is asked by Mihir sir. The question is, in a drug-drug co-crystal, sometimes quantity ratio of drug and co-former interacting are different, in which one concentration may be below onset of action.
Yeah. Yeah. What's the question? It's a statement, right? Is the question for?
Sometimes what happens that when drug and co-former both are APIs, and
Yes.
when they interact with each other to form a co-crystal, but sometimes may happen that if one concentration is below onset of action, which cannot give pharmacological action.
Yeah.
So in that case, what should be your comment, sir?
Yeah, I fully recognize that limitation. So that means the drug co-crystal has a limitation. We have to deal with it. Knowing is something we consider, but pharmacological activities are a must, right? So, of course, say if one drug has some properties need to be improved, which it can be improved by forming a co-crystal with another active. That's fine. You can form co-crystal to form to exhibit the improved property. Then you can maybe add another compound, excess compound as needed to it. It becomes a mixture of the drug co-crystal and another drug in order to satisfy the dose requirement. If the dose is one other than 100, it's gonna be very hard to do. But I think that it's a real challenge, and then maybe one main factor that limit the widespread use of this strategy. But you will find examples where this can be very nicely. They have nice one-to-one co-crystal, one to two co-crystals just happen to form to meet this requirement for those. So who knows?
Yeah.
I think if we develop the fundamental science, so when the situation when the situation arises, we are ready to do it. That'll be how we should we train students that they're equipped with knowledge. So when I go to work and develop some drugs, oh, we can do, take this strategy, then everyone will benefit from that.
That's true. True.
Thank you, sir. This will really show a new pathway to our researcher. Thank you. Now, due to time constraint, we are not able to address each and every questions. So the delegates are requested to please mail your questions to register mail ID, and then ma'am will forward that questions to sir. And when we receive the answers, we will mail you the same.
Sounds good. Yeah. I think this is recorded, right? The meeting recorded.
Yes, sir. The meeting is recorded.
That's good. Because I need to sign off of the meeting. I like to see the other speakers talk also. But unfortunately, it's kind of late for me from my side. I need to sign off. But if you can send me the link after the conference, I really appreciate that.
Yes,
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 sir.
definitely, sir. We will share the link.
Great.
With you.
Yeah, thank you. I forgot to say Happy New Year to everyone. Yeah, it's a pleasure to see everyone. Yeah.
You too. Very happy New Year, sir.
Okay. Yeah. Bye bye.
Thank you. Thank you very much, sir.
Thank you,
Thank you
sir.
very much.
Okay. Now we will move forward to our next technical session. In our second technical session, we have with us Dr. CVS Subramaniam, sir. Currently, the sir is professor and principal of Department of Pharmaceutics, GR College of Pharmacy, Osmani University. Previously, the sir has been associated with various colleges and university like the College of Pharmacy, JN Medical College, Bangalore, Birla Institute of Technology and Science, Rajasthan, College of Pharmaceutical Sciences, Manipal, Bapuji Pharmacy College, Karnataka. Sir has been obtained the BPharm and MPharm degree from College of Pharmaceutical Science, Andhra University, Visakhapatnam, and PhD degree from Birla Institute of Technology, Rajasthan. Sir is having an expertise in various areas like physical pharmacy, pre-formulation studies, novel drug delivery system, which can be reflected with the publication. Sir has more than 40 research publications, 15 books to his credit. Sir has guided more than 17 PhD students, more than 40 MPharm students, and 25 BPharm projects. Sir is also a referee of review of research articles for Indian Journal of Pharmaceutical Science. Sir is also a member of editor board of Indian Journal of Pharmaceutical Education and Research. Sir is also a lifetime member of various national societies like Indian Pharmaceutical Association, Association of Pharmaceutical Teachers, Indian Pharmaceutical Graduate Associates. Sir has received research funding from various agencies for the projects like a research promotion scheme by EICT, MODROP by EICT, Industrial Institute Partnership Cell by EICT. Sir has been recognized and awarded with various prestigious awards like Ranbaxy Research Award for Pharmaceutical Sciences and the Good Teacher Award. With this brief introduction, I would like to invite Sir to deliver his lecture on value addition strategies of pharmaceutical multi-component system.
Shall I please share? I will share with this one, scene sharing.
The participants are requested, please don't touch on the screen. Yes, sir, your presentation is visible now.
Oh, wait, let's see. Good morning, every one of you. I take this opportunity to thank the Saurashtra University Pharmacy Department for giving me an opportunity to share my thoughts with you. I have, looking at the distinguished speakers on this particular specialized topic, I took a medium role of blending some of the basic things with the amalgamating with multi-component systems. When I tried to frame this particular topic, I visualized that multi-component systems is having a value addition to the pharmaceutical products. Therefore, and the value addition of these products has not been covered in any of the syllabus of B pharmacy or M pharmacy. Therefore, it is a topic which can be combined easily with this. And then I have made a topic like value addition strategies on pharmaceutical products and multi-component system. Then I try to combine and giving some of my thoughts about the co-crystals, which is currently being interested by many people. So let me, this is the value addition pharmaceuticals and multi-component systems. The objectives of this particular session is to appreciate the meaning of value addition. When you call a particular product is a value addition, drug product and services, along with a few strategies. This is what I would like to do it, and then understand the concept and meaning of multi-component system. When you call it as a multi-component system, how it has to be visualized. Then try to criticize some of the current theories proposed in the formation of co-crystals, particularly theory aspects, and appreciate the issues that needs specialized knowledge and skill expertise, a technology of co-crystals at the university level, at the student level, to have much more understanding about these particular topics. So then when I start talking about value addition, all of us know how to add value to our products. There is no doubt about it. But what is being not done is we have not being taught in that particular fashion. For example, if you say that you have a tablet of a particular drug and somebody wants to have a rapid action of that particular drug, you have an alternative, that is an injection, a rapidly giving pain relief. So therefore, this is one aspect of value addition. Or if you can tolerate, you can avoid injection and take a tablet. This is a value addition, but we were not taught like that. Say, for example, you are taking 3 tablets per day. Today, you have a sustained release product. That itself is a value addition. But we have, therefore, what I want to tell you in this particular session is whatever knowledge we have acquired so far has been processed, streamlined, and focused in a particular direction where you can devote your direction to achieve that particular aspect. So this is what I would like to ask. Therefore, what I am talking about value addition in a basic level, everybody knows, but based on that particular thing, probably we may have to build up further. So this is how I have framed. When you talk about value addition, we always consider it as an improved addition. When I talk about value addition, I don't mean that you are going to introduce a branded product. It need not be an NDA. Even if a replacement of a brand innovator product into a generic form itself also can be called as that. But by practice with us, with the industry, we consider generic product development, ANDA, is automatic and implied in the act. Therefore, there is certain amount of innovation, but it is not being considered in that particular perspective. Now, let us see how value addition is connected to innovation. Or both are same or both are different. Therefore, we should be able to understand what is not a value addition, what is really value addition. So therefore, value addition is connected, need not include commercial opportunities. If you are going to introduce a product, it does not mean that it is commercial opportunities means cost reduction. You are reducing a product by cost reduction. Cost reduction is a value, no doubt about it, because patients perceives like that. But it is not being considered as a value addition for various reasons. If the price is a little high, even still, if it has got an advanced opportunities, advantages, then it will benefit user. So what is more important is, no doubt cost is a factor, but what is the extra benefit that he will get in purchasing a particular product. Therefore, cost reduction, though it is considered good, but it cannot be considered as a value addition. Does not depend upon expiration date. When all patented products are expired, are being expired at 20 years, then naturally ANDAs will come. So you are just mimicking one particular product to another, and also you are doing cost reduction. But again, that cannot be considered as a value addition, but because you are just making it equal, bioequivalence. Therefore, it cannot be called. It is not a substitute for the existing products, me too products, etc. So these things cannot be called as a value addition by default by our people. So therefore, value addition means go beyond certain expectations. You have expectations definitely will be met, but go beyond that particular one. That is where that small addition will give you a value, and this is important, and provide something more to the purchaser. Whatever he is paying, he gets, that's right, that is being assured. But what is that more you are doing, that is much more important than this. Therefore, value addition has got certain strings attached to call them. Basically, in today's context, when QBD has been introduced in the product development, any amount of developments that are being taking place in the product, it must be science-based. If it is a discovery, no doubt there will be inventive step, therefore it will become a patent. Even co-crystals are also patentable. So that is a value addition. But if it is not their inventive step, at least there is certain amount of innovation, then you can call it as a value addition. Something extra, features, more than expected. That means there is a shift from the traditional way of giving a pill and going above, that is beyond a pill. Therefore, the pharmaceutical industry, from sellers to a partner in the healthcare, by providing this extra value addition, is considered to be an important aspect. Technologically, we are strong, we will do a lot of developments, but how these things will finally reach the patient is much more important. Therefore, any value addition should be perceived as a patient-centric business model, and this has to be adopted by the manufacturers. Value addition products, medicines in Europe has mentioned that value addition criteria, you are having an existing drug, a new indication has been given to that particular, suggested for that particular drug. Yes, it is a value addition. Purpose, product repositioning. This repositioning is not an easy in marketplace. So it requires more innovation. No doubt it may be in generally generic market also. So innovation should be associated with a better differentiation of products. It cannot be called as me or me too products. That is where repurposing is required. Product expansion. What is meant by expansion? Today you have a product with capsule. You are making it a tablet. Tomorrow you will make this particular product as an ointment. Tomorrow, day after tomorrow, you may produce this particular product as an eye drops or ear drops. So there is an expansion of that particular product for various segments and various needs of the patients. Say, for example, you have a product, conventional product. Tomorrow it will become a controlled drug delivery system. There is an expansion. That is a value addition. That is why controlled drug delivery systems have got the patentability consideration in it. They need to be covered under the criteria of patents. What are the criteria of patents? Novelty, non-obviousness, and industrial utility. So therefore, when you go for value addition or novelty, whatever is it, it should have industrial utility. Today, what is the criteria for evaluating the utility? Bioequivalence requirement is essential. This may be already the product is available. It is not just equivalence. You have a product, one product, you are preparing an equal product. That is only a regulatory compliance. But what is more important in value addition is to prove improved activity. This is very important. So this will build up the better, what is called as better reputation and better margins. It was just a bit stuck up. Just let me see what. Yeah. Now coming to value addition products, let me see. Products. What you can call it as a multi-component system. That is what is me just talking now. It may be a physical form. Again, in physical forms, not all physical forms are multi-component systems. Say, for example, you have a polymorphic form. Only one form is there. Where it can be multi-component. You have a only metastable form. It cannot become a multi-component system by definition. So multi-drug systems, yes, multi-drug systems also can be considered as multi-component systems because there is a therapeutic synergism in that. Say, for example, your well-known example is antihypertensive drugs are combined with the diuretics. Yes, there is certain amount of synergism and getting a benefit, what is called as more than A plus B. And multi-ingredient systems, for example, excipients. A very good example is atorvastatin with calcium carbonate, which is being done by Lipitor. Until now, this calcium carbonate, what it does to atorvastatin, is there any replacement of calcium carbonate excipient, just excipient, will really take it to the level of this particular combination is still under investigation. This is a multi-component system. But what is carbonate? Carbonate is an excipient. Whether you take carbonates, take other carbonates, or you separate calcium and take other calcium salts or other carbonate salts, nothing has come nearer to this. Therefore, these are multi-value additions and what we are going to discuss today. What is the advantage of this? Reduce the risk of failure. See, for example, if you are attempting for a drug discovery, it takes a very long time and heavy expenditure. But if you take a small piece of these type of value additions, your expenditure is less, your efforts are less, your timeline is less. Therefore, what you can do is you can reduce the risk of failure. Failure will be less, and even if there is a failure, it will be less risky rather than going into the area of drug discovery and all this. Selection of drug safety and effectiveness. This is again a value addition because these two are connected with the patient. Whether it is when it becomes a safety, is it impurities associated with API or high and low ingredients present extra on that. Therefore, this how you decide the safety and effectiveness by design space using QBD. Therefore, that is the area where you have got a lot of choice to select what is to be selected, what is not to be selected. So you give this particular aspect for safetiness and effectiveness while selecting that. Selection of appropriate strength, type of API, that is physical forms or otherwise other forms, route, size, shape, etc. What is more important in all these aspects is compliant with the treatment regime. Anything that supports the compliance by the patient will give, or anything that adheres to the regimen gives certain amount of novelty and sustained release and improved therapeutic management. So these are the things. So therefore, value addition, technologically, we will do a lot on our own, but at the same time, we also have got the perceptions of a particular patient behind our mind when we are applying value addition. Patent demands, patient demand quality improvement, innovative products. Product alone is not sufficient. There are several issues that services combined with education seek support. So for example, supply of health cards like Apple Watch today, you have, it will tell you what is the pulse rate, what is the blood pressure, and several things it will give when you are walking. So therefore, from pharmaceutical industry point of view, if they want to go beyond the product and make their particular product innovative or what value addition, certain amount of complementary things are training programs, diagnosis camps, back office support practices, etc. These are required. And this is only not on the paper. It is being practiced elsewhere. Only thing is in India, we are not able to do it. But with the advent of PharmD, with the advent of pharmacists moving closer to the patients or closer to the doctors, this particular aspect will take care. But it has got a lot of difficulties also. But what is more important is this is being practiced. If we can try it, it can become, and this for practicing this, you require industry backup also. So value addition, citizens, value added services are necessary evil. It has to be done. It is expenditure is more because additional expenditure, additional manpower deployment is there. The job description will be expanded. It is therefore it is difficult, but it is a necessary evil. Value addition, let us advantages. Look for partnership with academia, healthcare organizations, hospitals, doctors, etc. Whenever we talk about this particular value addition, it is a business proposition. When it becomes a business proposition, then several factors will come. Whether this particular company we has to associate with A hospital or B hospital, or A corporate hospital or B corporate hospital, or A doctor or B doctor. How it is to be done? Because so therefore the local politics or local competition, local behavior, all these things will become an impediment for implementing this thing. This is at this particular stage, an educational institution which has got neither inclination to any of these sides, it can stand upright and offer its services, therefore liaison with the, at least to extend education and also training to the desired people, to the required people, are associating with the local corporate hospitals in types of services, camps, etc., will take these medicines, and the role of pharmacist nearer to the patients. It has got a limitations are also there. It has got a negative impact, particularly cost base, and also there it requires expenditure. Where do you have to focus your expenditure? Where you have to bifurcate your expenditure? How much you devote for these particular things? How other, because of this particular work, how the other activities gets diluted, etc. These are several questions that will be asked. But what is more important is the patient is not very much worried, looking at the company where it has come, but what is more important is what it has given to him in terms of what is worried about execution of this particular process. So innovative, there are 4 strategies I would like to talk about it. Innovators' point of view. a particular innovator is there, he will try to get all the benefit for 20 years, but what is being done is he always prevents competition by just taking several measures. Maybe cost is one particular measure, but extending originator's product exclusivity for elderly people, or exclusivity for children, or exclusivity, or something like that. They also combine their product, originator product, with something else, like lovastatin is used for what is lovastatin plus extended release niacin for hyperlipidemia. So like that, this is being extended. Therefore, evergreening the strategy is another. This is done by generics, and evergreening strategy followed by innovators, how they will keep their product live in the market, and how the generic people will challenge the evergreening strategy of that particular company. That is challenging the patent they will do. They will also go for combination of drugs. So therefore, they transform the generic products into a much more effective manner. These are called as two evergreen strategies. Each one is followed by another. Now, one is life cycle management is another strategy that is being used. Optimizing the brand product. How they will optimize? Of course, everybody will tell about cost containment. Another straightforward way is novel drug delivery systems. Or otherwise, optimizing primary and secondary manufacture. Until then, it is primary manufacture means API production. Secondary manufacture means formulation product. These two are separate organizations. So they have to optimize here also, and they have to optimize secondary manufacture also. So it so happened in one particular company, secondary manufacture, it is being covered under patent, and by agreement, primary manufacture, API fellow is supplying. When secondary manufacturer is getting all the benefits, he also expects primary manufacturer, API company also wants reap much more benefits from this particular one. Suddenly they said that we increase the API cost so that it will become, then only they will be able to supply. So what is the next? Then they have started cost reduction by these various methods. In this particular company, it tells that it took 80 years for them to optimize the costs, etc., to reduce, so that each partner in this particular situation can be equally benefited. And this is a case of branded products. Extend more mature molecules. Low business due to new molecules. What is meant by mature molecule? Say, for example, a drug is there. It is a branded product. As per law, they are getting all the benefits for 20 years. After 5 years, suddenly another molecule will come, and that catch up. Then naturally, this molecule will be less taken care by that. Therefore, all these are called matured molecules. So what they will do with these mature molecules? These molecules, because of competition, new molecules have come. Their molecule has gone into the low level, that is life cycle management. So they have to go for new treatment lines, because these molecules, low business due to new molecules coming in. Say, for example, everybody knows cimetidine is the molecule that has come in H1 receptor, H2 receptor antagonist. But cimetidine has got a very short life. Afterwards, ranitidine has come and picked up. So therefore, any particular product, though it is branded, it is not possible for the company to keep them alive. So therefore, lifetime cycle management strategy is very, very important aspect. And there are another important example is, today there is a drug, it is being claimed by one type of cancer, and as per act, they have to use that particular drug for that type of cancer only. But if you can find the same drug is useful for another type of cancer also, then it can also be taken permission from FDA, and then it can be used for that also. So if it is not being, that is how you have to look for mature molecules. What are the other offers? This is already patented molecules. losing market over a period of time. Brand loyalty made hospitals to prescribe mature molecules in hospital formulary. This is a situation where the molecule is going into low share, going out of market because of competition of new molecules. So in this particular case, this particular brand is being taken by a hospital and kept it in a hospital formulary. That means whatever this particular, for this particular treatment, this drug has to be compulsorily used by that hospital doctors for that particular treatment. Therefore, that is how, that because it is already branded, it is being used for a long time, it is known for safety. Today, new molecule had come into the market. So this is one example. This has happened in Paris, where they have taken that, they have included all these type of molecules based on their utility. They have put it in this, and of course there is life cycle management is a pricing strategy. Expansion strategies. This is what is called the reproofing. In our syllabus, B Pharm or M Pharm, reproofing is not being touched upon. One is reformulation, changing the dosage form, maybe once in a day, earlier, or reducing the side effects, extended release, or transdermal. So these are all this all pharmaceutics people will know. This is repositioning. Repositioning of thalidomide is a very, very novel example to tell. Already know about thalidomide tragedy. Thalidomide has gone, it was removed because of various toxicity levels. But today, thalidomide has come back again as an emergency treatment for certain type of diseases. What is more important is, when thalidomide is being taken by the patients, they have got the relief, they have got the intended one, but there are side effects, that is one. Therefore, now what had happened is thalidomide has come into the market again for various other specific indications, and it has got a life. That is another repositioning is Viagra. What is Viagra? How it has come? If you see in clinical trials, this particular one particular compound has been given to the human subjects, and it is for what is called as the sudden losing of control of urine. Say, for example, somebody coughs very high, suddenly urine leakage will be there. Somebody has done various other reasons, laughing, good laughing, heavy laughing is there. Suddenly he will lose his control on urine. So for this particular bladder control, this particular drug has been clinical trials. What it has got, whether it is successful or not, a different issue. But what the subjects have told the investigators is that it is extending the ejaculation, it is maintaining erectiles for longer time. And that is how this particular molecule. So what is being observed in a clinical trials, what are the things, certain things are very important, that will anyway, they will be on limelight, they will come. But what is more important, the other things. So therefore, extension strategies will come. And another example is many metabolites have become drugs later period. It is a classic example of repositioning. Reinvestigate also, there are several examples are there. So what I say is, this is one value addition. You start with a drug centric, a new indication, or a safer drugs, less expensive. Once you have got this particular strategy, move on to this. This is what is called as 1 minute. Repurposing. So how many strategies are there? Phenotype strategy, genetics, text, omics, essay, all these are strategies. By using innovative value-added strategies, you can get repositioning. Once you get this particular stage, you go back, it will become disease-centric, specific disease, rare disease, neglected disease, what all these type of things, they are being done. Therefore, this is a strategy whereby you can go for more innovation. Super generics and hybrid strategies are another type of strategies. The new platforms, new versions, platforms of IP, intellectual property creators. This is one platform. Another platform is a technology platform. Technology platform is a novel type of template by which you can remove one drug and put another drug. It will work. It will give you certain amount of data. This is platform. I don't talk about it. Platforms are slowly introduced in syllabus also. In B Farm also, there was some amount of platforms, technology platforms, etc. So shift from production to therapy, addressing clinical needs of the patient. Pharmacovigilance is again value added. It is not just prescribing drug, detection, assessment, understanding and prevention of adverse effects. This is another area. Then otherwise followed by generics, getting therapeutic advantages. This is important. Faster time into the market, low investment in open invitation and partnerships. These are few aspects, and this is what medicines of Europe has given 5 pillars. Patient needs, one pill fits to all is not correct. We have to meet unmet methods, quality care, access to high quality care, life cycle management, and after patent expiry also life cycle management. So these are various things that are being given, which gives you a bird's eye view of value addition. So all these things are not being covered in a particular syllabus, maybe in B Farm level. So these aspects are to be taught to the students, and the best way of explaining them or teaching them is by using this particular one, what is called as practice school. Practice school, 7th semester B Farm, and also for Farm D places. Somewhere something has to be done. Probably I felt that all the pharmacists are B Farm, M Farm, what is meant by value addition may guide them into a proper direction to get into that. With these few things, I will just move on to our multi-component systems. It is simple. It is 2 components are there, or more than 2 components are there. There is a supporting structure, but there is a interconnection. This interconnection gives you integrity. These are important aspects. In phase rule, of course, you have lot definition, B Farm textbook information. This is only a definition which gives you ice, water, the water and water vapor, 3 components, 3-phase system, but only 1 component. But most of the times, to our basic knowledge, phase rule has been done good with inorganic or metal substances, and slowly, as Dr. Calvin has informed just now, it has gone into organic molecules and multi-component systems. Though there are difficulties are involved, simple diagrams like eutectic mixtures we have studied in physical pharmacy, but when it comes to the solid, we have started, until now, we have seen only 2 extremes. One side is a drug, another side is co-farmer. What is the interaction between these 2? But today he has given one more line in between the graph and showed that this is a co-crystal. So therefore, our understanding of multi-component system is still to be developed in a large way. Actually, the multi-component system has been evolved, given that much importance because DMSO is used in the drug discovery to get in high throughput screening, to get a large number of drugs at a stretch for screening, and by various reasons, one type of other drug has come into picture and into life. Therefore, it is being done. This is how it is being. Both BCS class 2 as well as class 4 drugs have given much more importance in multi-component systems. There are various examples of multi-component systems. You have micellar systems, surfactant is used, monomolecular complexes, beta-cyclodextrins are used, Heterotropic solubilization, heterotrophs are used. Solid dispersions, carrier molecules are used. Co-crystals, co-formers are used. All these are this. But what is more important, a supersaturated drug delivery system is not a multi-component system. It is supersaturated, means only one component. If you add something, then it will become multi-component. That is how other people use HPMC or PVPK14 are being used. In solid dispersions also, you have etistic, solid solutions, glass solutions, amorphous precipitates, complexes, etc. All these things are there, and these are all textbook information. Just I want to bring it all these into one particular platform where we can say that they are also multi-component systems, including amorphous precipitates, glassy solutions with drug with polymers. So this is what I have mentioned about it. Now what US FDA will tell about co-crystals. Now I have come to this particular one. Co-crystals have become much more important, and this is the reason why even in second year B Pharm physical pharmacy also I have introduced co-crystals as a type along with the salts as this based on their therapeutic superiority. So now B Pharm onwards, the way B Pharm student will study BCS classification, in a similar manner, co-crystals are also being studied by B Pharmacy students also. So it is a crystalline material, that is what US FDA says, API and co-former it says, and single crystal lattice, that is what is important as per the definition. All co-crystals means definitely they are solid state, there are no doubt about it. And some cases they are solvents, but only thing is solvent is replaced by another co-former. As per US FDA, lack of ionizable groups is the rule of difference. When it is ionizable groups, then it cannot be considered as a co-crystal. That is what it says. The differences, how it decides, it is based on the differences in pKa value. This everybody knows, most of the people. Stoichiometry 1:1 or 1:2. Again, this is important criteria. That means this particular ratio, whether this will what is called as reversible, whether these two components will dissociate, release the drug for higher solubility, gets absorbed or not. This is to be studied in dissolution. It can assume the status of NDA or new drug applications or ANDA. Co-crystals of two drugs US FDA considers as a fixed dose combination. It is because two drugs are there, both are drugs. For the purpose of classification, it is considered as a fixed dose combination and it comes in NDA. Now let us see some of the theories. People say that hydrogen bonding propensity is required for having a loose contacts or reversible weak contacts. And already we know Lipinski rule. Lipinski rule, if you see, hydrogen bonding acceptors more than 5, hydrogen bonding the donors more than 5, like this rule of 5 is being there. If we know this particular knowledge of hydrogen donors and acceptors, can we use this for include into developing the theories for co-crystal formation? You have chemistry softwares are there, these are being used. Mole inspiration, ChemSketch gives you a lot of physicochemical data. If you take this particular data, you can come to a reasonably good conclusion where to start and how to start. And synthon supramolecular architecture, Kelvin has shown homosynthones or synthones. This gives you only prima facie evidence. It will not give you a figure, what is like pKa A minus pKa B is less than 3. That type of conclusions are not there. It gives you pairing, how these two molecules are interacting. It gives you evidence how packing is possible, but there is no numerical indication, only qualitative. So this can be used as a parameter in the checklist. Fabian methods based on molecular descriptors. In fact, M-Pharm pharmaceutics people also will study about computer-aided drug development. Lot of molecular receptors or descriptors are there. These descriptor databases are available even for pairing of drug and co-pharma. Certain amount of correlation and two aspects are being shown. Shape and polarity are the two criteria that can satisfy Fabian. Next comes Hansen solubility parameter. See, solubility parameter delta is a cohesive charge density, cohesive energy density. This cohesive energy density is applicable for liquids. For solids, it is not that much easily applicable. And the method of preparation, of course, you can do fragmental constants, but it is a long process, but it gives you a roughly good some indication. But what is more important here is not the cohesive energy, it is the adhesive energy that is required. There is another crystal, virtual crystal screening. So these are the technologies that are being used. Now, is really co-crystals, say solubility, say dissolution, are they really work in vivo? Whatever you are doing in vitro, are they really work? Then there is an assumption. Crystals are expected to improve, co-crystals are expected to improve bioavailability. What is that particular bioavailability guideline? Is it soluble throughout the GIT tract? Is it soluble in HCl? Is it soluble in intestine? What is the guiding principle for you? And this is known as spring and parachute action. If you see time and concentration, if you see normal product, this is spring. A crystalline drug will have low bioavailability, and this is being done by co-crystals. First one is spring. Suddenly the dissolution, the drug concentration increases. After that, after reaching the peak that should be able to maintain for longer period. This is what is called as spring parachute effect. So at the initial point of view, any co-crystal that you administer, you should get this spring parachute effect. But this spring parachute effect will not sustain forever. Therefore, in the first 1 or 2 hours, if you can get this particular spring and parachute effect, first 2 hours all drug will be absorbed. Most of the drug will be absorbed. Most of the problems will be solved. Therefore, the co-crystals and bioavailability has been by spring and parachute concept, where you should be a rapid, high dissolution, high absorption, at least even for a short period of time. And if you take only co-crystals, if you further add excipients, etc., they also improve. Some of the co-crystals can form supersaturated solutions. But these supersaturated solutions, what is the guarantee? They don't get precipitated in the body. So therefore, hydrophilic polymers, HP, I'll take a few minutes and then. Now, what is the theory? What is solubility? What is crystallization? Solubility means drug going into solution. Crystallization means drug coming out of the solution. Therefore, if you look at the interaction between solute, solvent and crystallization, the same phenomena, same interaction will happen. For solution also same interaction. You add heat, it will dissolve. Interaction remains same. You reduce the temperature, the precipitation, crystallization will come. Otherwise, the interactions. Therefore, solute-solvent interactions giving much more differences in this particular phenomena is not being taken care. When you talk about Mears crystallization theory, how exactly these crystals are being formed? You have a supersaturated solution, a nucleation, crystal growth. All these are the theories. Again, much of these theories are applicable to inorganic substances. But what is more important in Mayer's crystallization theory is what are the factors that will decide the crystallization. Say for example, in a co-crystal, they are in solution. You have drug is also there, co-former is there. Now you have to get a nucleus. Tell me, drug will form nucleus, nucleation, or co-crystal will form, co-former will form crystals. By looking at this solubility behavior, drug will first form a nuclei. Next, complementary sites are available, then solute will form. Once co-former forms, then co-former's complementary is required. So drug will form. Therefore, what you can imagine during crystallization, what is happening is, to me it appears, first drug will form, then co-former will form, then again drug will form. This is what is called as layerization. That is what Kevin has shown you, that this is the type of arrangement. This is basically from theories we have got it. Now, issues related to specialization. Today, a particular industry preparing co-crystals as API, because API, I am telling you, API mixture, European Medicines Agency decided co-crystals are API mixtures, defined as that. This particular things, today you say for example, omeprazole. Omeprazole is unstable. You have to get entry-coated omeprazole. Today in the market, several industries are there. They will supply entry-coated omeprazole as raw materials. And formulation department, formulation, that means secondary manufacture, they take that particular entry-coated material and do the tableting and other things. Therefore, in the entire work activity, co-crystals as API should be a specialized aspect, and it should be done as a primary manufacture. In the secondary manufacture, that what we do, the way we get API from various companies, we get co-crystals from that particular company, do further processing. So for this reason, excipients has become equally important as API. That means it itself has developed into a material science. So a combination of primary manufacture and secondary manufacture is a win-win situation where both will benefited. Otherwise, a single individual, it is difficult to handle. Now, skill. Do we have sufficient skill? Say for example, today I will ask my student, you prepare a salt. Can he go to the laboratory and prepare a salt? All our students, all of us will do large number of organic molecules, big molecules, derivatives, this, that, synthesis of sulfonamides, synthesis of acetanilide, all these things we will do. Or they have got the experience to prepare a salt? Less. This can be done in practice school. Because that is basically important, because what we think about co-crystal is less stronger than salts. Less strong, first you should start with salt, then you will get. This is one aspect which we are lagging, which has to be developed. Second one is, when you take two molecules, one co-former and another is a drug. This drug is, for example, base. When you ask for this company to do that, it will prepare a salt, send you a salt. So all weak acids are available as sodium, potassium salts. All bases are available as hydrochloride, sulfate salts. You take these raw materials and mix them. Several counter ions are there. What these counter ions will do in the formation of co-crystals? Again, it is a guesswork, because we know a strong base will precipitate a weak base. A strong acid will precipitate a weak acid. So these aspects you know. But what is more important is all these counter ions or co-ions present when you get raw materials from other companies, what these have got a role to play. Again, some light is being thrown by Kevin, but what is to be done is, what is your raw material? Can you prepare, as a chemistry person, a base by itself? Many times all bases are liquids. They pass on HCl, so they form HCl salt, then they will get. Issues related to technology. There are some of these issues. What I would like to talk about in particular thing in a high dose products, because you have a drug and you have co-pharma. When you are combining this, it is possible that the size of the tablet or size of the dose may increase. When such a cases, how can you handle? This is major problem with that. So in that particular situation, while imagining this particular situation, I am of the opinion 3D printing, which has just been included in the syllabus and also being upgraded and also being approved by US FDA for one particular product, can be an ample opportunity to provide. This is one, this is a traditional type of thing. 3D again, I'm not talking of 3D printing. This is the entire one. It can do powder, it can do extrusion, it can do liquid. All these type of aspects are possible in 3D printing. Therefore, this can offer a solution when there are technological problems, particularly weight related aspects. So there are several problems with these co-crystals, etc. Scalability, stability problems, bioequivalence issues, processing. Therefore, what is more important here is a huge amount of chemistry inputs are required. A huge amount of technology inputs are required. These two hands has to be joined. Then only we will be able to reap the benefits of co-crystals. Thank you very much. At this particular stage, I will stop and I will hand over the mic to.
Thank you, sir. Thank
You
you for.
Yes,
sir. Yes, sir.
Right. Okay. Thank you very much.
Thank you, sir, for enlightening us with your vast knowledge and experience. Sir, you really shown us the new side of the coin, that is value addition strategies of pharmaceutical multi-component system with respect to patentability and utility. Now, sir, we have some questions that is asked by our participants.
Can
I
just trying to get me what I have to do to see the faces?
Sir, you have to stop sharing your screen.
Stop sharing my screen.
Yes.
Okay, I have done stop. No, wait. Now tell me, madam. Thank you very much. Because sir, I am also.
So can I read the questions that is asked by?
Yeah, if possible, I will answer because I am not a scientist. Just to imagine physical pharmacy background and how imagine things. Right? Okay, tell me.
Yes, sir. The first question that is asked by Yashika Patel. What are the essential activities of pharmaceutical supply chain quality management?
Madam, difficult for me to tell this particular madam. Supply chain. I don't have any idea. I will answer the next one.
Okay, sir. The next question that is asked by Sejal Kuman. The question is, if two drugs used as a co-crystal, how can we find they both are compatible with each other or not?
No, of necessity, these two drugs should be one is acid and another is a base. This is one. If they are going to form a co-crystal 1:1 or 1:2, again there is a limitation of dose. When all these things permits, this is possible. I tell you, co-crystals means how it looks like. See, if you have two painkillers are different. See, one drug which has got a side effect has to be handled by another drug, and there is a possibility of forming a co-crystal. So side effects, this is what we tell terminology in quality, we tell no, intended, unexplained, undescribed effects. Quality is defined as to give intended purpose as well as unintended, undefined. That under analgesics it comes as it should not cause gastric ulcer is the one condition. But nobody will write it will not cause gastric ulcer. So therefore, in a situation like that, it is possible, but there are limitations about that particular dose. One has to be very, very careful when he is going for these type of things. because you never know stoichiometry where it will work.
Thank you, sir.
Right.
Now the next questions that is asked by Professor Dr. A.K. Bansal, sir.
Right, sir.
How can pharmacy students more access to computational software as they are very costly? Can we create a cluster to share resources so that more students can work on these concepts?
Yeah, that is true. Actually, what we are doing is, I am now working on online open source softwares. From that, I have downloaded dissolution software and solver software. These softwares have become once upon a time very difficult, but I am trying to do in that, and all these things will be shared by all the students. And I am developing exercises also and bring them into laboratory manuals, because now I am 65, I am no more principal. So I devote my time in the laboratory and improve the skill of the students, particularly practical skill. So some of the laboratory manuals in future will give you a large number of open source free downloadable softwares, particularly dissolution, PK solver, and also what is called as optimization, factorial designs. These type of things I am looking, so that these resources can be available to the students to learn. But it can be certainly a possibility. We can have a common platform and share all these type of things. Fine. Thank you very much, Bansal sir.
Thank you, sir. Thank you very much, sir.
I have
Now,
taken a very long time, I think, to explain things. Or many
No.
things are being done in that. Okay, right. Speak.
Thank you, sir. Now, there is one announcement that we will have a short tea break that is of 10 minutes. So, sharp after 10 minutes, we will assemble over here for the next technical session.
Thank you very
Thank
much,
you.
every one of you, for your patient hearing. I did not have so many odd things underline this, that, etc. because not habituated. Because COVID forced us to learn all these things. So thank you very much.
Thank you, sir.
We'll meet again, right, after 10 minutes.
Yes, sir.
Fine.
Thank you, sir.
Okay, 11:50. I hope 11:50.
Yes, sir. Yes, sir. At 11:50.
Right, right. Okay.
Now again, welcome you all in this international e-seminar. Now in our third session, we will have Dr. A.K. Bansal sir with us. Currently, the sir is professor and head of Department of Pharmaceutics at National Institute of Pharmaceutical Education and Research, Punjab. Sir earned his master and doctor degree in pharmacy from University of Delhi. Sir worked as a senior scientist and a group leader in JK Pharmaceuticals and Ranbaxy Laboratory Limited for 8 years. Sir is having an expertise in various areas like pre-formulation profiling, solid state characterization, improvement of aqueous solubility, enhancement of oral bioavailability, and compaction physics. Sir has more than 170 research publications, 27 review papers, 11 granted patents to his credit. Sir and his research work has completed more than 550 industrial projects. Sir has guided more than 100 M.Pharm students, more than 15 PhD students. Sir has also received the grant from various granting agencies like ICMR, DEST, Department of Biotechnology. Sir has been also recognized and awarded with various prestigious awards like a Fellow of American Association of Pharmaceutical Scientists, Distinguished Educator and Research Award by American Association of Indian Pharmaceutical Scientists. Sir is also appointed as an editor board member in various journals like Journal of Excipients and Food Chemicals, Journal of Pharmaceutical Sciences, Molecular Pharmaceutics, and many more. With this brief introduction, I would like to invite Sir to deliver his lecture on co-crystal as a novel solid form for pharmaceutical development. Over to you, sir.
Madam, sir is not able to unmute.
No, I think just now I am able to unmute my mic today about a topic.
Okay.
So
Okay,
I think I have shared with you my recorded talk so that there is no technical problem during the lecture. So I would like to thank the organizers, especially Professor Mihir Rawar, for inviting me to this very pertinent conference on a topic which is very relevant in these times. As has been highlighted that biopharmaceutical properties like solubility, compressibility, hygroscopicity, and overall processability are as important as the potency of the drug. So sometimes there is a need to modulate these properties using the classical approaches that were used for solid form selection. Co-crystal has opened up an entirely new area for utilizing the supramolecular chemistry skills to improve the biopharmaceutical properties. So I thank the organizers for this wonderful event, and you can now play the recorded lecture that I had sent you, and I will be available for any questions thereafter my lecture is over. Thank you very much. Hello everyone. I'm going to talk to you today about a topic entitled co-crystals as novel solid form for pharmaceutical development. The other speakers have already talked about various types of multi-component crystalline systems and the protocols that are used for screening of these multi-component systems. My emphasis is going to be on another topic. If we have chosen the co-crystal as the solid form for development, what are the challenges that are involved? First, in identifying the most optimal co-crystal candidate, and then subsequently conversion of the co-crystal to a final drug product like a tablet or a capsule or any other dosage form. During the drug discovery stage, as we all know, the pharmaceutical scientists are trying to optimize various properties like potency of the molecule, its selectivity, pharmacokinetic properties, and the material properties. By the material properties, we mean some of the important physicochemical properties like aqueous solubility, intestinal permeability, stability, compactability, and processability. We have the choice of picking up from different solid forms the most suitable one for our pharmaceutical development. Salt forms, as we know, can only be used for the ionizable drug molecules. Crystalline form can be used, amorphous form can be used. Another new area for the selection of the solid form has been opened up by the field of co-crystals. The co-crystals can be developed for non-ionizable molecules, and they can provide us significant solubility benefit while maintaining a reasonable level of physical as well as chemical stability. In today's talk, we will try to understand that if we have generated a number of co-crystal candidates for further development, what is going to be the criteria for choosing the most optimal co-crystal candidate and the challenges that are involved in its subsequent conversion into a solid dosage form. This is going to be the flow of the presentation. We'll briefly talk about different solid forms of the API. What are the triggers that encourage research in the area of co-crystals? Strategy for selection of co-crystal based on the pre-formulation studies. And we will take an example here or a case study to exemplify this. The formulation development using the co-crystal, and finally we'll try to draw some important conclusions. This picture shows the significance of solid form in pharmaceutical development. Solid form can influence properties like solubility, dissolution rate, stability, both chemical as well as physical, hygroscopicity, mechanical properties like flow and compactability, and overall processability of API into the finished dosage form. An ideal solid form should possess all these properties, and they overlap in this zone, which is called as the sweet spot. This is the zone which has the optimized properties for developability, and our emphasis during the selection of the co-crystal candidate, or for that matter any other solid form, should be to pick up a form which provides us a balance of all these properties that are important for the finished dosage form. At the molecular level, the solid form of the API can be classified broadly into two categories, crystalline and amorphous. Crystalline solids could be multi-component or they can be single component or multi-component. In multi-component, we have hydrates and solvents, salt form, and co-crystals. Our emphasis today is going to be on co-crystals. Co-crystals can further exist either as anhydrous non-solvated co-crystals and their polymorphs, hydrated solvated co-crystals and their polymorphs, co-crystals of salts and their polymorphs, solvates and hydrates. On the other side, in comparison to the crystalline form, we can also use the amorphous form. But today's talk is going to focus on the area of co-crystals. Co-crystal has been defined by the scientific community as solids that are crystalline, single-phase materials composed of two or more different molecular and or ionic compounds, generally in a stoichiometric ratio, which are neither solvates nor simple salts. US FDA also defines the co-crystal on pretty much the similar line, except for the fact that they mention that they exist in a defined stoichiometric ratio within the same crystal lattice that are associated by non-ionic and non-covalent bond, which means the molecules in the crystal lattice are held together by non-ionic and non-covalent bond. And this is what differentiates from the salt forms. Here is an example of a co-crystal, carbamazepine and succinic acid system. Carbamazepine is the API and succinic acid is the coformer. In the crystal lattice, we can see there is participation of both the API and the coformer. Obviously, we have a large number of co-crystals that have been reported in the literature for many APIs now. Co-crystals differ from simple salts, solvents, and other solid form, and this picture tries to highlight that aspect. The key differences in co-crystals and the other solid forms are: it is a multi-component system, unlike the polymorphs. Unlike the salt, there is no complete transfer of proton involved in the case of co-crystals, and its synthesis does not require presence of the ionizable functional moiety. Co-crystals have definite stoichiometry, unlike the solvents and the hydrates. Second component in the co-crystal, that is the coformer, is non-volatile in nature, unlike the salts and the hydrates. This table shows the comparative overview of co-crystals with other solid forms in respect of various parameters like solubility, dissolution rate, physical stability, chemical stability, compaction properties, hygroscopicity, and the challenges during the scale up of the process. In the case of polymorphs, there is a 2 to 3 times improvement in solubility that is possible, while in the case of amorphous, it is 4 to 14 times. Salt forms can give 2 to 1000 times improvement in the solubility. Co-crystals can give an improvement in solubility of 4 to 20 times. In terms of the solubility advantage, they are comparable to the amorphous form, but additionally they have physical stability, which is much better than the amorphous form, since amorphous form is the high energy form and has a tendency to convert back to the crystalline form. All the solid forms, since they improve the solubility, will also enhance the dissolution rate. In terms of the physical stability, co-crystal physical stability may be challenging, while the amorphous form is always challenging because it is the metastable form. Chemical stability for polymorphs gives us low to medium risk. For amorphous, it is high risk. Here it is low risk, and in the case of co-crystals, it may be challenging. Similarly, for other properties, we have compared the various aspects of the polymorphs, amorphous form, salt form, and the co-crystals. What comes out from this table is that although most of the time we are trying to optimize on one of the biopharmaceutical property like the solubility, the other aspects of the solid form may not be encouraging for conversion it into the final drug product. So if we have to choose the optimal solid form like a co-crystal for the further development, we have to look at all these properties simultaneously and pick up one co-crystal candidate which provides us optimal properties in terms of all these parameters. We cannot simply look at one of the property and compromise on all other, because all other properties can also affect the safety and efficacy of the drug product. The interest in the area of co-crystals is increasing day by day. And we can see in year 2015, there were 660 publications and 297 patents which were filed in the area of co-crystals. Amongst the patients, 80% of the patents were filed by the pharmaceutical industry, while 20% were filed by the academic institutions like universities. This clearly shows that there is a lot of interest in the industry for converting the APIs into co-crystals and then using it for conversion to the final dosage form. We now come over to the important part of our lecture today. Here we'll talk about a pre-formulation guided strategy for selection of the co-crystal candidate for drug product development. We are going to use a pre-formulation flowchart so that from various co-crystal candidates, we can pick up the most important one. This will also help us in understanding what pre-formulation properties are important when we select a co-crystal candidate. This is the flow diagram which we published as part of a review article that was published in Journal of Pharmaceutical Sciences. If we have to pick up the co-crystal candidate, the first thing that we need to see is the safety of the co-former. The co-former that has been used for creation of the co-crystal should not be unsafe to use in a pharmaceutical product. Also, the weight contribution of the co-former to the overall co-crystal should be within limits, because otherwise it will be difficult to convert the co-crystal into a final drug product, especially for high dose drugs. The first most important aspect that we need to evaluate is that of the solubility, and within that we will also look at the eutectic constant determination, because this tells us that what is going to be the duration of the solubility advantage during the dissolution stage. pH solubility profile, solubility in biorelevant media, phase transformation profile are all important components of the solubility studies. Thereafter, we should look at the dissolution, intestinal permeability, physical and chemical stability, solution state and photostability, and the hygroscopicity of the co-crystal candidates. Thereafter, you need to ask a question: are these critical parameters satisfied by the co-crystal candidate? If the answer to this is yes, then you can move over to the next stage. If the answer to this is no, you will have to go back and choose other co-crystal candidates. Thereafter, we evaluate other properties like polymorphic screening of the co-crystal candidates, the mechanical properties, the compatibility of the co-crystal with the excipient that are going to be used in the formulation, what is the microenvironment pH of the co-crystal, and what is going to be the impact of this microenvironment pH both on the solubility as well as the stability of the drug product. If these properties, which we call as the additional pre-formulation studies, are also satisfied, then we ask another question: are above parameters satisfied? And if the answer is yes, then we will select the co-crystal candidate for further product development. But if the answer to this is no, then we can have two possibilities. Can we use rational formulation development and container closure system or process design to mitigate the challenges that have been identified? If that is possible, then we'll use those strategies to develop the final product. But if these seem to be inadequate to address the challenges, we'll have to go back to the selection of the optimal co-crystal candidate. And obviously, then again, we'll have to look at the critical pre-formulation studies and additional pre-formulation studies for the new candidates that have been selected. So this is a flow chart, or you can say a decision tree that can be used for choosing the best co-crystal candidate from amongst the number of co-crystal candidates that are generated due to the screening of the co-crystals. As you must have always seen that when we do the screening of the co-crystal, we end up with a number of co-crystal candidates. But out of them, which one is going to be the most suitable for formulation development needs to be assessed on a scientific basis. We now move over to a case study wherein we will take up rivaroxaban as the drug candidate and talk about co-crystal based drug product for improving the biopharmaceutical performance of rivaroxaban. Rivaroxaban is a drug candidate which has solubility and dissolution rate limited oral bioavailability. These are the calculations of the deabsorbable and the time of dissolution, which proves that for the 20 milligram dose, solubility and dissolution rate are the limiting factors for oral absorption. The innovator for rivaroxaban has addressed this issue through hydrophilization of the micronized form of rivaroxaban using fluidized bed granulation for the 10 milligram strength. But unfortunately, using this approach, the innovator could not solve the problem of poor bioavailability of the 20 milligram strength, which is the higher strength that is also available. The required value of solubility to improve the bioavailability of 20 milligram dose was calculated as 11 micrograms per ml, while the current solubility of rivaroxaban is about 7 to 8 micrograms per ml. So we are looking at doubling the aqueous solubility of rivaroxaban so that the bioavailability of the higher strength, that is 20 milligrams, can also be optimized. As per the decision tree, we proceeded with the experimental evaluation. The first aspect that was assessed was the safety of the coformer. 3 coformers were subsequently chosen, as we'll see in the next slide. The malonic acid, oxalic acid dihydrate, and 4-HBA. And these were then evaluated in terms of their quantity that is going to be present in 20 milligram strength of rivaroxaban. And then we evaluated whether they are within the limits that are existing in the approved products or in certain other guidelines of the regulatory agencies. As is very clear, for malonic acid, the quantity present in the co-crystal for 20 milligram strength would be 7.5 milligram, whereas 80.1 milligram has already been approved in other products. Similarly, for oxalic acid, it would be 11 milligram, and for 4-HBA, it is going to be 19.3 milligrams. So all these were within the range as approved by the regulatory agencies previously. So the safety of the coformer is not going to be an issue. This was the screening protocol that was used for the selection of the coformers. We started with 28 coformers, and while choosing this, we adopted the delta PK rule, which says that it should be the difference of delta PK should be less than 0 for co-crystal formation. And additionally, synthon approach was adopted, and the synthon structures that were identified for formation of the co-crystal with rivaroxaban were oxazolidinone, aminomorpholidone, and carboxamide. Thermal behavior of the binary mixtures of the coformers with the API was assessed using the DSC at different heating rates, and this was the decision tree that was adopted to choose the coformer. And if a co-crystal was formed, that coformer was chosen and taken forward. But if it led to the formation of the eutectic, we did not take that coformer further. And as I already told you, these were the 3 coformers that were selected: malonic acid, oxalic acid dihydrate, and 4-hydroxybenzoic acid. We then went ahead with assessing the essential properties of the pre-formulation profiling, and we identified solubility, dissolution, permeability, stability, and hygroscopicity as the essential properties. Solubility was the primary objective for which we were creating the co-crystals, hence it had to be evaluated very, very critically. And once we change the solubility, we must also identify its effect on dissolution, permeability, stability, and hygroscopicity. And it should not happen that in the attempt to improve the solubility, we are compromising on some of the other properties, because that is going to overall affect the safety and efficacy of the final drug product. The solubility of the co-crystals were evaluated in biorelevant media, that is the fasted and the fed medium. So in this particular bar chart, we have shown the values of solubility that were obtained. So this is the solubility that was obtained by rivaroxaban in the fasted medium and in the fed medium. This is with the malonic acid co-crystal, this is with the oxalic acid, and this is with 4-HBA. As you can see, 4-HBA did not lead to any significant improvement in the solubility. Hence, it would not be an ideal candidate for taking further. However, we kept on evaluating it for other parameters as well. And from this, it is apparent that the malonic acid is giving us the highest water solubility and hence could be a probable candidate for further development. However, we must ensure that in an attempt to improve the solubility, properties like hygroscopicity are not being compromised. So in the subsequent slides, we will evaluate the other pre-formulation parameters of these co-crystal candidates. The pH solubility profile of the malonic acid and oxalic acid co-crystals were evaluated. And a very important aspect that came out of this particular study was the phase transformation time, which was more than 4 hours for the malonic acid co-crystal, but it was only 20 minutes for the oxalic acid. This means that the solubility advantage that is going to be obtained from the malonic acid is going to be like a spring parachute effect, and the solubility is going to be maintained at a higher supersaturated level for biopharmaceutically relevant time frames. While in the case of the oxalic acid, the higher solubility that was obtained falls back to the lower solubility very, very quickly, and this is called as a spring effect. The dissolution studies were carried out, and in this also the rank ordering was that rivaroxaban had the least dissolution rate, while the malonic acid co-crystal has the highest dissolution rate. The studies were carried out using a dissolution medium which had a zinc index of 0.45, so that we were able to discriminate the dissolution profile from the various co-crystal candidates. The permeability of the rivaroxaban across CACO2 cell lines was also evaluated, so that we could assess how the coformer affected the intestinal permeability. It came out from the study on the rivaroxaban alone that it has a significant PGP efflux parameter, as the ratio of apical to basolateral and basolateral to apical was found to be 1.88. For a compound with a efflux ratio of 1.5 to 2, a positive effect of the inhibitor indicates the implication of the efflux transporter. This means that rivaroxaban is a substrate for the PGP efflux transporter. Then we compared all the co-crystals and their physical mixtures, and we found that there was no significant difference in the apical to basolateral or basolateral to apical permeability of the co-crystal candidates, which means there was no impact of the co-crystallization of the on the permeability of rivaroxaban. The solid state stability and the effect of microenvironment pH was then assessed. There was no significant degradation in all the samples in the solid state after 1 month at 40 degrees Celsius, 75% RH in the closed and moist conditions. No significant degradation in all the samples in solid state when they were stored at 3 months at 40 degrees Celsius and 75% RH in open and dry condition. However, in the presence of a basic stressor, that is sodium carbonate, the rivaroxaban 4-HBA co-crystal showed a yellow discoloration with 50% reduction in the assay of 4-HBA. However, there was no evidence of degradation of the rivaroxaban. Now, this leads to a very interesting scenario that although the rivaroxaban was not degrading, the coformer was degrading in one of the conditions. And if the coformer degrades, obviously there is going to be the disturbance of the crystal lattice, because the original co-crystal was formed with 4-HBA, while during the stability it has degraded into some other compound. The hygroscopicity studies were carried out for all the co-crystal candidates using dynamic vapor sorption technique. The 4-HBA co-crystal only showed 0.03 weight gain, which is shown by this red colored line, and this black color line, which is almost overlapping, is for the rivaroxaban. This blue colored line is for the malonic acid co-crystal, wherein you see a significant water in uptake has been shown by the sample, especially at higher RH values. On this axis, you have the relative humidity values, and this green color profile is for the co-crystal with the oxalic acid, which shows a steep weight loss of 8.8% at RH values below 20%, owing to the loss of the water molecule from the co-crystal hydrate structure. The co-crystal that was obtained with oxalic acid, in fact, was a hydrate form, and when you store it below the 20% RH, it used to show a steep loss of 8.8%. So the rank ordering of the co-crystal candidates in terms of their hygroscopicity was that malonic acid co-crystal was the most hygroscopic one, followed by oxalic acid, 4-HBA, and rivaroxaban, which was almost equal to the 4-HBA. 39 times higher water sorption was exhibited by rivaroxaban malonic acid co-crystal as compared to the neat API, which can be a cause of concern because hygroscopicity can complicate the processing of the co-crystal into the final dosage form. These are the separate sorption and desorption profile that have been shown. So this is malonic acid alone. The red one is for the co-crystal and the black one is for the rivaroxaban. We in fact then went ahead to understand the molecular basis for water sorption behavior of rivaroxaban malonic acid co-crystal, since it was showing 39 times higher water sorption as compared to the pure API. We adopted a crystallographic based approach to understand the molecular behavior, wherein the first was to identify the surface groups that are exposed on various solid forms that were tested. This is the crystal structure of rivaroxaban, which was picked up from the literature. This one is the arrangement of the molecules in the co-crystal with the malonic acid. In the case of rivaroxaban, we have anti-parallel arrangement of two rivaroxaban molecules interacting through a bilateral NHO hydrogen bonding to form a dimer in AB-AB fashion. And these were the characteristics that were observed in the case of the co-crystal. The next study was the phase indexation study, which is done using a single crystal XRD machine after generating a single crystal. And this was done for all the three samples, rivaroxaban, pure malonic acid, and their co-crystal. We generated very useful information through this particular exercise, and we identified the crystallographic planes or the facets in each sample. What was the relative contribution of these facets to the surface groups that were exposed on these facets, and what was the nature of these surface functional groups. Were they hydrophobic in nature? HPHO denotes hydrophobicity, while HPHI stands for hydrophilic groups. And this was done for rivaroxaban, malonic acid, and riv-mal co-crystal. We also identified the groups that were exposed on the surface using the Mercury software and looking at their crystallographic information file. What are the groups that are exposed on the surfaces of the samples? So this is for rivaroxaban, this is for malonic acid, and this is for the co-crystal. The third step was recognizing the nature of the chemical groups that are exposed on the surface, whether they are hydrophilic or hydrophobic. because if the groups are more hydrophilic in nature, their interaction with water is going to be much higher as compared to the hydrophobic groups. This was done using the computer software, wherein we are able to see the relative hydrophilicity and hydrophobicity of the various groups that are present in a chemical structure. So we did it for rivaroxaban. The blue colored zones show the groups which are relatively hydrophilic, and the brown ones are the groups which are hydrophobic in nature. So these are the hydrophobic groups of rivaroxaban, while these are the groups which are relatively hydrophilic in nature. We calculated the molecular lipophilic potential using the in silico method, and this was the malonic acid molecule. As you can see here, most of the molecule is hydrophilic in nature, as it is blue colored throughout the molecular structure. Then we calculated the relative contribution of hydrophilic, hydrophobic, and the combination of both to the facets. And we came up with this interesting observation. In the case of the malonic acid, this is the contribution from the hydrophilic groups, while for rivaroxaban, this is the contribution. So majority of the contribution is coming from the hydrophobic groups. While there is nothing which is completely hydrophilic, this zone indicates the facets which have both hydrophilic as well as hydrophobic groups. While in the case of the co-crystal, we can see that the hydrophobic groups have reduced from 65.2 to 57.9, while the combination of hydrophilic and hydrophobic groups had increased from 34.8 to 42.1. This could be one of the reasons for the higher hygroscopicity of the co-crystal as compared to the rivaroxaban. So we concluded that the enhanced hydrophilic plus hydrophobic contribution from 34.8 to 42.1 and reduced hydrophobic contribution from 65.2 to 57.9% could be one of the contributors to the enhanced hygroscopicity behavior of the malonic acid co-crystal. However, the difference in the hygroscopicity of rivaroxaban versus the co-crystal was 39 times. And the values that we obtained in the previous case, these are only marginally different between rivaroxaban and the malonic acid co-crystal. So from 65, it is going to 57, and from 34, it is going to 42. So this does not explain in completeness the reason or the molecular reason for the higher hygroscopicity of the co-crystal with the malonic acid. We then went on to understand that the strength of the hydrogen bond or the strength of the interaction between the hydrophilic groups in the co-crystal and water could be an additional parameter that is contributing to the increased hygroscopicity profile. This was studied using the DFT calculation, which is the density function tool, and the hydrogen bonding strength was assessed by calculating the interaction energy between the various molecules and water. In the case of rivaroxaban, the interaction with water is associated with an energy of minus 5.1, while for the malonic acid, the interaction with water at this particular COOH group has a much stronger interaction because it has a lower energy of minus 10.2 kilocalories per mole. Similarly, the interaction of malonic acid with water at this position, that is the carbonyl position, also comes up with a stronger interaction with respect to the rivaroxaban and water interaction. So presence of surface chemical groups that have strong hydrogen bonding interaction with water molecules favors the water sorption tendency of crystalline solid as compared to those with weak hydrogen bonding interactions. So what came up from this study was that the amount of water sorbed by the co-crystal is governed by the surface molecular environment and additionally by the strength of the hydrogen bonding between the surface groups that are exposed and the water molecules. So this is the entire study in a nutshell, that we first evaluated the nature of the facet in terms of the hydrophilicity or the hydrophobicity. What is the relative contribution of these to each facet, and what is the strength of the hydrogen bonding interaction between the groups that were exposed on the facets and the water. This work has been published in Molecular Pharmaceutics. Then we went ahead to evaluate the other desirable properties in the pre-formulation study, which included the polymorph screening, mechanical properties, microenvironment pH studies, and the compatibility studies with the excipients. There were no polymorphic forms obtained for the co-crystal with 4-HPA and with malonic acid. The co-crystal with oxalic acid exists as a tetrahydrate form. Anhydrous form of oxalic acid co-crystal could be formed by storing the co-crystal at a RH value less than 20%, which was very obvious and could be seen in the DVS studies that we discussed earlier. However, at the ambient humidity, the anhydrous form gets converted into the hydrate form. The mechanical properties in terms of the tabletability were evaluated. This is a low critical pre-formulation parameter for low dose drug like rivaroxaban, because the dose that we were working on is only 20 milligrams. Nonetheless, we evaluated their compactability profile and we found that the co-crystal with malonic acid has a reasonably good tabletability, and it gives us the value which is higher than the 2 megapascals. So this is the tensile strength which is optimally required to form a good quality tablet, and we were getting a value which was higher than this. The microenvironment pH studies were carried out because they can affect both the solubility behavior as well as the stability behavior of a compound. A thin saturated layer of water is formed at dissolving surface of the solid particle, and this represents the microenvironment, and the pH at this region is called as the microenvironment pH. Rivaroxaban is a non-ionizable drug and has same microenvironment and bulk pH. So since it is a non-ionizable drug, hence the effect of microenvironment pH was not observed in solubility and the stability studies. So this again came out to be a non-critical pre-formulation parameter for rivaroxaban co-crystals. Subsequently, the compatibility studies of the co-crystal with the excipients need to be carried out, because you are going to use certain excipients for the development of the solid dosage form, which was a tablet in this particular case. We adopted a very conservative strategy here, and we used the same set of excipients as were used by the innovator in the original rivaroxaban drug product that is marketed in the international market. Because of this, the co-crystal excipient compatibility was not carried out with the co-crystal candidate. However, this should not be viewed as a general statement, because sometimes the excipients that are compatible with the drug may not be compatible with co-crystals, because there is likely interaction between the co-former and the co-crystal and the excipient. What comes out of this is that in the case of co-crystals, the compatibility has to be assessed not only with the API but with the excipient as well. We then adopted a weighted criteria matrix based on Lean Sigma approach to identify the best co-crystal candidate. These were the critical parameters that were identified: solubility, stability, hygroscopicity, permeability. And a weightage was allotted to these parameters. Solubility was given a weightage of 40, stability 30, hygroscopicity 20, permeability 10, and the total of these would be 100. And based on this, a net score for the 3 co-crystal candidates was calculated. We ended up with a net score of 480 for the malonic acid co-crystal, while it was 400 and 280 for oxalic acid and 4 HBA co-crystal. And the rank ordering of these co-crystal candidates with respect to their optimum nature for further dosage form developed was: the first rank was obtained by malonic acid, the second one by oxalic acid co-crystal, and the third one with 4 HBA co-crystal. Based on this, we can conclude that the malonic acid co-crystal was the best one for carrying out the further development into a solid dosage form. And we then developed a prototype formulation for the co-crystal with malonic acid. We developed various prototypes using the excipients that are shown here. The formulation was a tablet. The tooling was 6 millimeter convex tablet tooling. Process was a direct compression process. These were the in-process quality control parameters. The target DT was comparable to the innovator, that is 4 to 5 minutes, and the compression was carried out using a rotary tablet press. These are the quality control parameters that were obtained for these prototypes. So as you can see, the hardness was well within the acceptable ranges. Disintegration type was slightly higher in this case and in this case, while it was optimal in this particular case because we wanted a disintegration time of less than 5 minutes. The friability was acceptable in all the cases, and these are the tablets that were obtained for these 3 prototype formulae. Critical challenge that was identified for formulation development and the subsequent interventions that were decided was hygroscopicity, because we have chosen the co-crystal with malonic acid as the optimal candidate for formulation of the tablets. But we know from our hygroscopicity studies that this co-crystal was much more hygroscopic as compared to the API. Therefore, it was challenging to process this using a unit operation involving higher amount of water, such as wet granulation technique. And that is why we had to proceed with alternate manufacturing processes like dry granulation or the direct compression process. This is a general discussion on some of the suggested formulation strategies for overcoming the challenges that are posed by co-crystal during the formulation development. Some of the co-crystals will show very fast transformation to the API, which will be reflected as a spring effect. And in those cases, we will have to delay this conversion of the co-crystal to the API if we have to harness the solubility benefit from that particular co-crystal. This can be done either by using pH modifying agents or using surfactants, using polymers, or a combined approach involving surfactant and polymer. These are targeted at delaying the supersaturation from the co-crystals so that the benefits that we want to obtain from the solubility of the co-crystals can be obtained. For enhancing the dissolution, the particle size reduction can be used as one of the strategy. If permeation is causing the problem, then we may use some of the permeation enhancers, although this is not a very desirable strategy because most of the permeation enhancers will have safety related issues. They are toxic in nature. For improving the chemical stability, pH modifying agents can be used, and for improving the physical stability like the hygroscopicity, We can use desiccants in the formula like silicon dioxide, or we can choose a suitable manufacturing process, just like we did in our case, that instead of the wet granulation process, we chose a dry granulation or a direct compression process. The manufacturing environment can be controlled by controlling the relative humidity, and we can adopt a packaging strategy using packaging materials like aluminum cold form blister instead of the normal PVC based blisters. Microenvironment pH can be modified by adding buffers into the formulation. Compaction mediated phase transformation is sometimes observed in the case of co-crystals, and there we have to choose either a alternate formulation. For example, instead of a tablet, we may develop a capsule dosage form, or we may have to select a suitable cushioning agent that prevents this conversion of the co-crystal into the API during the compaction stage. The above suggestions can be applied to improve the product performance of co-crystal based product when the selected co-crystal candidate does not possess optimum physicochemical properties on its own. What is coming out on and what is getting highlighted is that although we were able to get a co-crystal with the enhanced water solubility, but some of the other pre-formulation properties suffered negatively, and then we have to adopt certain formulation based interventions to optimize those properties that got negatively affected by co-crystal formation. We then went ahead to perform an oral bioavailability study with the co-crystal and compared it to the innovator tablet, Xylarto. We used the powdered dosage form to enable administration to the rats. These were the plasma concentration that were obtained from the co-crystal tablet and powdered innovator tablet. As you can see, there is a very significant improvement in the oral bioavailability from the tablet that was made using the co-crystal with malonic acid. These are the pharmacokinetic parameters and their comparisons. There was a 6 times improvement in the Cmax. The Pmax reduced by 0.7 times, and there was almost a 5 times improvement in the area under the curve in the oral bioavailability study that was conducted in rats. To summarize the co-crystal product development, which means a formulation development using the co-crystal as the candidate, we first need to define the quality target product profile as per the principles of the quality by design. We then screen the co-formers. In this case, we had screened 28 co-formers, and then out of that we selected 3 co-formers. The safety of these co-formers was evaluated. In fact, this is a very important aspect which is ignored at many instances. The co-former that is used for formation of the co-crystal may not be safe for pharmaceutical usage. If we are looking at developing a drug product from that co-crystal, the safety of the co-former must be ensured right in the beginning, because later on it is going to give problems with the regulatory approval process. This is followed by the pre-formulation profiling of the candidates or the co-crystal candidates that were shortlisted. Rivaroxaban 4-HBA co-crystal was rejected or not taken forward because of poor stability. The co-crystal with the oxalic acid had a rapid transformation to the pure API, which means the solubility advantage was not maintained for biopharmaceutically relevant time frame. It also showed poor stability because we saw certain degradation occurring in these cases. Based on this, the co-crystal was selected as rivaroxaban malonic acid using a weighted criteria matrix. We then developed the formulation using the optimal co-crystal candidate and obtained improved in vivo performance and improved manufacturability. These are the take-home messages from today's presentations. Co-crystals have provided a broad platform for modulation of material properties that are critical during formulation development and have emerged as a promising alternative to traditionally used solid forms like polymorphs, salt forms, and amorphous form. Despite the rising number of research publication and patents, the systematic approach for development of co-crystal based formulation is lacking in the field of co-crystal, and we tried to fill this particular gap with this study that we carried out with rivaroxaban. Translational development of the novel solid form of co-crystals and its conversion from bench to bed may not be a straightforward process due to unique properties and structural features of the co-crystals. As outlined using the case study of rivaroxaban, the suggested pre-formulation based strategy can guide the product development in number of ways. It allows us to choose the suitable co-crystal for further development, helps in identifying potential problems during processing and manufacturing of the formulation, allows understanding of potential stability during the product shelf life, and also helps us in devising alternate strategies for handling, formulation, storage, and manufacturing of the final drug product. Thank you very much for your attention. If you have any questions, I will be happy to answer that. Later on, also, you can contact me through this email ID, akbansal@niper.ac.in. Thank you very much.
Thank you, sir. Thank you for giving an information regarding the pre-formulation aspects of co-crystal and industrial perspective and the significance of co-crystal. Now, sir, we had certain questions. Can I read that question, sir?
Yes, please go ahead.
Yes, sir. The first question that is asked by Mihir, sir. Formulation of co-crystal to improve the properties must focus only on physicochemical properties by industry, because by improving the physicochemical property, if mechanical property is destroyed, what will be the next step to be taken by industry?
Thank you, Professor Mihir, for that question. In fact, most of the time, the reason why we are developing co-crystal is to address the poor aqueous solubility of the APIs. In the process of improving the aqueous solubility, we may compromise on certain other properties like tabletability or the mechanical property, as Dr. Mihir has highlighted. In those cases, we have to look at certain aspect. If the dose of the drug is less, then most of the tabletability is going to be contributed by the excipients, not by the API or the co-crystal. In those cases, we don't have to worry much. In other cases, we have to evaluate that can this poor mechanical property be resolved by choosing the appropriate excipients or not. If it can be improved by using the appropriate excipients like microcrystalline cellulose or lactose monohydrate, then we can go ahead with the product development. But even if we are not able to get the good tabletability, we may have to drop that particular co-crystal candidate and look for some other alternate co-crystal candidate. That is what I was trying to highlight through this entire presentation, that we should not only look at one property that we were trying to improve. In the attempt to improve that property, we are likely to change all other properties also. And that is where the role of pre-formulation comes. So a very important question, and all people and students who are working in this area should keep this particular aspect in mind.
Thank you, sir. So the next question is, can we add any ingredient to increase the permeability of co-crystals?
Yes, we can add permeation enhancers, but we must again keep in our mind that all the permeation enhancers will have safety related issues because they modulate the normal biochemical processes of the cell layer. So you must keep this thing in mind that if you use a permeability enhancer, you should not end up in certain problems with the regulatory agencies. Your product may be rejected because the permeation enhancer that you have used may not be safe to use in a drug product.
Thank you, sir. Now, sir, the next question is, what is the role of microenvironmental pH study while preparing co-crystal?
It is important from various angles. The microenvironment pH can change the stability behavior of the API. The microenvironment pH can change the solubility behavior of the API. The interesting aspect is that not only the contribution of the API, but the contribution of the co-former to the microenvironment pH must be studied, because both of them can have an impact on both the stability as well as solubility of the API from the co-crystal.
Thank you, sir. Now almost all the questions are answered by sir. Thank you, sir.
Okay, thank you very much. I once again thank Dr. Mihir and all the organizers from Saurashtra University for organizing this wonderful conference. Thank you very much. I took a little more time than what was allotted to me. Sorry for that.
Sir. No, sir, it was really an informative session, sir.
Thank you, sir. It was very informative session. Thank you very much.
Thank you. Thank you.
Thank you, sir. Now we'll proceed for our fourth technical session. And for this, we have with us Professor Dr. Renu Chadda. Currently, the name is Professor, Institute of Pharmaceutical Sciences, Punjab University, Chandigarh. Ma'am is having an expertise in various areas like pharmaceutical chemistry, pharmaceutical analysis, which can be reflected from the publications that ma'am have more than 80 publications in various reputed journals, 3 patents to her credit. Ma'am have guided more than 7 PhD students and also provide a consultancy services to pharmaceutical industries. Ma'am have received grant awards and fellowships from various agencies like DST, CSIR, UGC, and ICMR, worth rupees more than 1.5 crore. With this brief introduction, I would like to invite ma'am to deliver her lecture on development of multi-component solid forms using crystal engineering approach. Over to you, ma'am.
Very much for the introduction, but I must correct you. I think you have taken my very old biodata. At present, I have guided 21 PhD students and 4 are still working with me. And the grant have been received by me is more than 3 to 3.5 crore. I think you have taken my very old biodata. Yeah.
Sorry,
anyhow, it's okay.
Sorry, ma'am.
Actually, yesterday I have sent my recent biodata also. I don't know from where you took my biodata. I don't know. Anyhow, it's okay. It's okay.
Sorry, madam, for that.
Students have done PhD with me.
Thank you, ma'am, for correcting me. And
no,
sorry, ma'am.
It's okay. It's okay. So I'll share the screen with you. I think it's okay.
Yes.
It's okay.
If you can take it from, yeah.
It's okay.
But it is the last slide.
Yeah.
Yes.
Yeah.
It is visible.
Please.
Yeah.
Sorry.
Unable.
Okay. Now we can start now. Now I'll talk about now first I must thank the organizer for inviting me to have a lecture. I couldn't hear the previous lecture by Dr. A.K. Bansal, but whatever I have heard, I think it was also on the co-crystal. My main aim will also be to the co-crystal. Anyhow, I'll talk about the multi-component solid form using crystal engineering approach. So now I will talk about the multi-component solid form, crystal engineering and designing of co-crystal, case studies of co-crystal, drug multi-component solid form, and then the conclusion. Now, we all know that drug development is a new chemical entities are predisposed to poor aqueous solubility that lead to dissolution limited bioavailability. Now, around 70 to 90 of the drugs in the development pipeline show poor pharmacokinetic profile owing to the lower water solubility. A recent study conducted on 812 drug candidates from AstraZeneca, Eli Lilly, GlaxoSmith, and so many showed that poor pharmacokinetic and bioavailability have remained the third most common cause of failure of the molecules in phase 1 clinical trial. Therefore, there is a challenging task to develop effective strategies to overcome these issues for the pharmaceutical industry. Now, cost-effective and time-saving approach is to modify the API without breaking or creating new chemical bonds in development of multi-component solid forms using crystal engineering approach. We don't have to break and form the new bond, but we will have a new entity with that are sustained by the hydrogen bond only. So the multi-component solid forms emerge as an innovative strategy to enhance the performance of active pharmaceutical ingredients without affecting their chemical integrity. So now what are the various types of multi-component solid form? Not only the co-crystal, but the multi-component solid form, it can be drug-drug co-crystal, it can be simple co-crystal, it can be co-amorphous, it can be a salt, or it can be a drug-drug eutectic. So while we are dealing with a multi-component form, we have to be very sure whether it's a co-crystal, it's a co-former, it's a salt, or it's a eutectic, because though they are all multi-components, but they have some different properties also. So first of all, in our labs, we have to be very sure that whatever we are prepared, we are mixing the two components. It can be the same, but there is a possibility that we may get the co-crystal, another possibility that we may get the eutectic. We may not get the co-crystal, and sometimes even the eutectic is taken as the if all the analytical techniques are not performed properly, then it is, and if we say it is not a co-crystal, sometimes it is a eutectic also, and many times we get the co-amorphous also, and many times instead of getting a co-crystal, we get a salt. When there is a proton transfer, we get a salt, but it seems to be the co-crystal. So we have to be very sure whether which multi-component form we are dealing with. Because if you want it has to be patented, we have to be very sure about the form. So first is the co-crystal. But our main focus is many times the co-crystals. The co-crystals are the solids that are crystalline single phase materials composed of two or more different molecules and are ionic compounds, generally in a stoichiometric ratio, which are neither solvent nor simple salts. So now, they shouldn't be salts. According to the FDA latest guidance on co-crystals, these are the solids that are crystalline materials composed of two or more molecules in the same crystal lattice. So, co-crystals are of interest in both academic research and pharmaceutical industry, in particular, as they represent the opportunity to assess new solid forms and therefore modified properties like solubility, dissolution, and their bioavailability. Now, a good example is provided by the structure of synthetic beta C aryl glucoside published by Deshpande and co-workers. Now, in this case, the crystal here in the crystal structure contains water, ethanol, a deuterionic component, and a neutral API. So, could be described as a co-crystal hydrate, solvate, deuterionic salt, or all of the above. It is the mixture of all. So now, for academic purposes, an acceptable co-farmer must be that something that can be purchased relatively easily. For pharmaceutical application, the list is more likely to be restricted to those compounds that are regarded as safe for the human consumption, that generally regarded as safe for the grassroots. So the FDA also has an additional list which is compiled by the Select Committee on Grass Substances. This list is maintained to analyze the health impact, the potential future risk from increasing dose of compounds on the grass list. So now this is a very small introduction to co-crystal, but possible alternatives to the co-crystallization. These are the co-amorphous. As I have told you that we have an intention to prepare a co-crystal, but we don't get a co-crystal. Instead, we get a co-amorphous, and this has been done in our own laboratory. We have faced this problem. Amorphous. So what are we? Everybody know what are amorphous solids are. Generally glass with short range molecular order, with no long range order of molecular packing or well defined molecular conformation. Whereas the co-crystals, they have a well defined structure. So co-amorphous drug formulation approach is characterized by the combination of two or more low molecular weight components that form a homogeneous amorphous single phase. So higher molecular interaction, higher conformational flexibility, and molecular level mixing. And this is our drug. This is our co-amorphous drug. Now, similarly, presence of more enhanced molecular mobility in amorphous material leads to higher reactivity, consequently resulting in thermodynamic stability compared to their crystalline counterparts. So because of their high thermodynamic energies, these solid forms offer solubility advantage as compared to the crystalline solid forms. So many examples are reported in literature utilizing this approach to improve the drug solubility, such as repaglanate, saccharin, naproxen, proline, indomethacin, arginine, and ritonavir, quercetin. These are the examples I have taken from the literature. Now, another possibility to the co-crystal is the eutectic mixture. The eutectic mixture. Now, what's eutectic? Eutectic is a mixture of two components, either drug coformer or the drug polymer, that are completely miscible in the liquid state, but immiscible in the solid state. They are held together by homomeric molecular interaction at specific molecular ratio. So it means the eutectic, it's a continuous solid solution. Then we'll say it is a solid solution, and it is a discontinuous solid solution. Then it is called as the eutectic. So therefore, when these are the eutectic and co-amorphous and the salt, these are the possible alternative to the drug component, to the multi-component solid form. So eutectics are multi-component crystalline solids as binary composition. So eutectics are formed between components that are non-isomorphous and are sustained by relatively weaker adhesive interaction. So self-self interaction and molecular shape mismatch between drug and the co-former lead to the eutectic mixture, whereas no self-interaction and molecular shape match between the drug and the co-former lead to the co-crystal. So classical example of the pharmaceutical eutectic is a mixture of lidocaine and prilocaine used as local anesthetic. So now anyhow, this is just an introduction to this. The focus of this talk is on the development of co-crystal using crystal engineering approach. So what's crystal engineering? Designing crystalline material for specified applications via manipulations of intermolecular interaction has been established as the field of crystal engineering. Now, the crystal engineering is defined as the understanding of intermolecular interaction in the context of crystal packing and the utilization of such understanding in the design of new solids with desired physical and chemical properties, leading to the changes in the bioavailability. So now, the concept of crystal engineering can be applied to various fields such as coordination polymers, nanotechnology, polymorphism, chiral resolution, purification, discovering new supramolecular synthesis, and in the pharmaceutical sciences for improving active pharmaceutical ingredients. So the crystal engineering teaches us how to bring molecules together exactly as we want. Can we engineer the molecules? Yes, we can engineer the molecules. We can bring them the way we want. So the field of crystal engineering related to pharmaceutical industry is poised to address two of the key issues, solubility and bioavailability, and has matured into a paradigm for the supramolecular synthesis of new compounds with the desired property. So now, as I have told you that my topic of the talk is the designing of the co-crystals. So first we must design the co-crystals and then we should prepare it. Now, in order to get a desirable co-crystal product of an API, the first step is to study the structure of the target API molecule and find out the functional groups which can form intermolecular interaction with the suitable co-former. So we have to see what is the structure, what do our API has the functional group which can form the hydrogen bond, because the co-crystals, they are sustained by the hydrogen bonding. So if our API doesn't have any group which can form hydrogen bonding, then there is a possibility that we may not get the co-crystal. Now, a detailed understanding of the supramolecular chemistry of the functional groups in API, which may form robust molecular synthesis such as acid-acid, acid-pyridine, acidamide, amide-amide, amide-pyridine, N-oxide, OHO, OHN, NHO, NHN is a prerequisite for designing a co-crystal because it facilitates the selection of the appropriate co-former. So for selecting the co-former, the first thing is that it should be of the Graf status. It should be safe for consumption. And besides this, a good co-farmer should be able to form heteromeric intermolecular interaction with the target molecules that are more favorable than the homomeric interaction that may exist, because there are intramolecular interactions are also there. So our co-farmers should be such so that it may overcome the intramolecular interaction and it should be interact with our API. So the two main types of supramolecular synthesis are there. One is the supramolecular homosynthesis and then supramolecular heterosynthesis. So supramolecular homosynthesis are the between the same complementary functional groups, carboxylic acid dimers, amide homosynthesis. So if you want a co-crystal to be formed, these have to be broken. They have to be broken. Only then they can form hydrogen bonding with the API. And then supramolecular heterosynthesis. They are between different but complementary functional groups. Acid pyridine heterosynthesis. So these are the typical hydrogen bonding in the crystal designing. These are the heterosynthesis. We can see that these are the various heterosynthesis between the amide, carboxylic acid, and so on. So now, how can we prepare the co-crystal? What are the various methods for preparing the co-crystal? Methods of preparation, solution method, mechanochemical method, and then there's the melting method. Various methods. First is the solution method. In the solution method, we have slow evaporative methods and reaction crystallization. In the mechanochemical method, we have solid state grinding and then the liquid assisted grinding. So we can use any of the methods to prepare our co-crystal. In solution crystallization method, either a solvent or a solvent mixture with similar solubilities for both the components are selected and stoichiometric amount of the reactants are used. It is called as the slow evaporation method. Or a solvent with different solubilities for both the component is selected and non-stoichiometric amount of the reactants are used, and that is called as the reaction crystallization method, and then we can get the co-crystal. In the next solvent drop grinding method, that's called as the green chemistry approach, because here we don't use much of the solvent. It involves co-grinding the components in stoichiometric ratio, co-grinding the components in the stoichiometric ratio under ambient conditions with addition of small amounts of suitable solvent to accelerate the co-crystallization. Okay. Now, again in the mechanochemical method, nowadays twin screw extrusion is another mechanochemical method in which two parallel screws are rotated in the same or opposing directions, facilitating the mixing and movement of materials. It is a continuous and scalable process, making it ideal for the industrial purposes. Twin screw, because then we can have it made in the bulk. Now next is the hot melt extrusion for co-crystal formation is a thermal microscopic method which allows the melt profile of two component system to be mapped and ascertain if a molecular complex or co-crystal phase is present versus system with simple eutectic profile. So we have to see during the course of the reaction that whether we are getting a eutectic or we are getting a co-crystal. Next is the ultrasound assisted slurry crystallization have been used in screening and preparation of the co-crystals. That if it's very difficult to prepare the co-crystals, then we can use the ultrasound assisted, and then we can pass ultrasound to the slurry of the both the components, and there's a possibility that we may get the co-crystal. Nowadays, we can have even we can in the microwave also in the microwave synthesizers can also be used to prepare the co-crystal. So now after the preparation, the very important part is the cook is the characterization. As I have told you that there is a possibility of the alternative salt formation, eutectic formation, or there is a coamorphous. So what are the various methods that can be used for the characterization of the co-crystal? In our laboratory, we are very particular about the co-crystal because if we are preparing a co-crystal, then we have to be sure that it is a co-crystal. It shouldn't be any eutectic. Of course, if it is eutectic, and it should be eutectic, it should be reported as a eutectic. Eutectic should not be reported as a co-crystal. I have seen so many papers that they have not characterized it properly, and even the eutectic has been reported as a co-crystal. So the characterization is by thermal analysis, spectroscopic method, and then the X-ray detection. Thermal analysis, hot stage microscopy, of course, it's not very important. The most important is the differential scanning. That the first technique that has to be used is the differential scanning calorimeter. We have to see what's the melting point of the co-crystal. It should be between the coformer and the API. Then thermogrammetry. Sometimes the solvent may be formed. Thermogrammetry, and then once we are sure that it has a unique temperature melting point other than the coformer and the drug, then we should go in for the spectroscopic technique to see infrared. IR is very important. We have to, because if co-crystal is formed, there are some perturbation, there are some changes in the vibrational frequency of the functional groups. Because if hydrogen bonding is there is some changes in the vibrational frequency of the functional group, which are responsible for the hydrogen bonding between the drug and the coformer. And then there is a solid state NMR. Not NMR because co-crystal are the properties of a compound in the solid state. Once it will go to the solution, they are supposed to be dissolved. So we have to characterize our co-crystal in the solid state. Therefore, we have to see the solid state. So solid state NMR is also a very good technique to characterize our co-crystal. Then X-ray photoelectron spectroscopy, then X-ray diffraction, powder X-ray diffraction, and the single crystal X-ray diffraction. So now this is the ultimate technique for the co-crystal characterization. Of course, in the powder X-ray diffraction technique, we will see that there are some changes in the new diffraction peaks will be appeared in the co-crystal, and some characteristic peaks which are present in our drug and the coformer, they will disappear, and new characteristic peaks will be appeared. But the ultimate method is the single crystal, because with the help of single crystal, we can see that hydrogen bonding is there, and actually our co-crystal has been formed. In case, sometimes it's very difficult to have a single crystal. And therefore, with the help of powder X-ray diffraction method, we can determine the structure. And this we have been doing it in our laboratory with the help of a software, material studio software. We can convert the two-dimensional powder X-ray diffraction to the three-dimensional structure, and we have been doing it in our laboratory also. Software is there. So first, I'll give you the example of hydrochlorothiazide and nicotinamide co-crystal. This we have been prepared, and it has been, and we have submitted a patent, and now we have granted the patent, Indian patent number. My students and myself here, hydrochlorothiazide and nicotinamide. Now it inverts the cost-effective method for increasing the efficacy of a hypertensive drug, hydrochlorothiazide. It has prepared to achieve an enhanced aquasolubility and subsequently the bioavailability. So it showed 8-fold aqueous enhancement in the solubility in comparison to the pure drug achieved within the 30 minutes, with substantial increase in the hypertensive efficacy in DOCA salt-induced hypertensive graphs. Now the stability of the co-crystal at 30% did not show any significant changes in the PXRD pattern as well as the solubility value. It was found to be stable, and determination of LD15 female vista rat indicated it to be a safe with no death and any sign of toxicity even at the dose of 200 mg per kg of the body weight. So how it was prepared? So the novel crystal of hydrochlorothiazide with nicotinamide was prepared by recrystallization method, and 148.9 milligram of hydrochlorothiazide were dissolved in 10 ml of acetonitride pre-saturated with nicotinamide and allowed to evaporate slowly at the room temperature. Now the crystals suitable for single access diffraction were obtained in 3 days, filtered and dried under vacuum at 30 degrees for 2 days and stored in an airtight glass vial. So this is the PXRD pattern. We can see this is the hydrochlorothiazide and this is the hydrochlorothiazide X-ray. Appearance of unique refraction at 2 theta value of 5.8, 12.9, 6.1 and 23.9 and 25 in the co-crystal, different from the drug and the co-pharma. This is the DSC scan. Hydrochlorothiazide, and this is our nicotinamide, and this is our co-crystal. This is our co-crystal in between the two. This is in between the FTIS spectra of the shifting of carbonyl stretching frequency of hydrochlorothiazide from 1694 to 1597, shifting of NH frequency from 3368 centimeter inverse to 3376 centimeter inverse, inferred that the carbonyl group and NH group are participating in the hydrogen bonding. Absence of peak corresponding to NH3 moiety in the region of 3100 to 2500 centimeters shows that both the drug and co-pharma are present in the neutral state. No salt formation, no salt formation. That is very, very important. So this is a single crystal X-ray that shows that space group orthorhombic space group. These are the single crystal structure of our co-crystal, and actually we can see the hydrogen bonding between the drug and the co-pharma. That confirms the formation of the arrangement of, now we can see the arrangement of hydrochlorothiazide molecules in unit cell in crystal structure of palm oil. So we can see that this is the hydrogen bonding between the drug and the co-pharma. Drug and the hydrogen bonding between the drug and the co-pharma. And this is the arrangement of drug and co-pharma in the unit cell. Now The solubility of hydrochlorothiazide, nicotinamide, and hydrochlorothiazide were performed in 0.1 normal HCl and the deionized water at 25 degrees in water bath shaker. Samples were analyzed and the samples were analyzed were assessed for drug content at 316 nanometer, eightfold increase in the solubility in 0.1 normal HCl and fivefold increase in the distilled water was observed in comparison to the pre-drug. And the stability study, the DSC scans of the multi-component crystals of hydrochlorothiazide after storage under accelerated stability conditions shows marginal changes in the DSC scan. That shows that our coke was stable also. The evaluation of the anti-hypertensive activity of hydrochlorothiazide and its co-crystal was performed in the DOCA salt-induced hypertensive female, that's 200 to 300 grams weight. The treatment of hypertensive rats with hydrochlorothiazide and the co-crystal led to significant attenuation of the elevated systolic blood pressure level of the DOCA salt-induced hypertensive rats in comparison to the DOCA control and vehicle control. Now, the enhanced hypertensive activity of the co-crystal is attributed to the increased solubility of the drug when hydrogen bonded to the highly water-soluble nicotinamide. Now, the next example is our oxcarbazepine. This is also our patent, and this patent have also been, we submitted in 212, and now it has been patented by the patent number 183. Again, this is to the typex. The oxcarbazepine is anticonvulsant drug used in the. This is our first patent. This is our first co-crystal which was patent, and after that we have sent the hydrochlorothiazide. Now we have submitted so many patents, and we are getting them patented also. The oxcarbazepine is an anticonvulsant drug used in the treatment of epilepsy. This motivated to explore the potential of oxcarbazepine to co-crystallize with an aim to enhance the solubility and dissolution profile and in turn the oral solubility. So methods of again. So we have prepared oxcarbazepine with succinic acid with acetic acid were prepared by the solution method, and then oxcarbazepine and saccharine was prepared by fast evaporation method. Two methods were used. So these are the just photographs. Now these are the DSC scans of thermograms of oxcarbazepine co-format and their multi-component form. Oxcarbazepine, and this is the oxcarbazepine acid co-crystal. Then there is the oxcarbazepine saccharine co-crystal, only single peak, and then there is the acetic. Of course, there is some water of solution was there. This is the single melting point. Hot stage microscope, and then these are the X-ray pattern of oxcarbazepine and its multi-component form, oxcarbazepine, succinic acid, and their base co-crystal. Single unique peak, and then saccharine and oxcarbazepine and saccharine physical mixture, and this is the co-crystal. Of acetic acid and the dissolution, and then because there was some water solution, water or solution was there. Then after dissolution, we get this, and after the post dissolution, after it was dissolved, we get the C. So now again we have got done the single, the solid state NMR study was also done. Now, in the solid state NMR of carbazepine and saccharine, main spectral perturbation appear in the amide carbon. Now, actually, in the solid state NMR, gate so the peaks are very broad. If we take the solid state NMR of hydrogen atom, so therefore, it was we could take the solid state NMR of carbon. So we can see that the groups which are the carbon to which the groups are attached responsible in the hydrogen bonding, there was some perturbation was seen on those groups also. So in the case of carbazepine succinic acid, main spectral perturbation was appeared in amide carbon of both oxcarbazepine and saccharine, and saccharine, and in C22. You can see the C22 species which is adjacent to the SO2 group of saccharine. So these oxygen atoms, they are responsible for the hydrogen bonding with oxcarbazepine, and the shift therefore there's a shift in this carbon, which is the most likely interacting both amide carbonyl as well as the SO2 of saccharine in the oxcarbazepine. Similarly, in the C13 resonance characteristic of acetic acid molecules, C23 and C24. These are the groups which are responsible for the hydrogen bonding, can be seen clearly in spectrum of the oxcarbazepine and acetic acid. So these are the crystal structure. The oxcarbazepine succinic acid was obtained from the solution as crystal of 2 table size, whereas in saccharine and acetic acid, we could not get the single crystal, but we could prepare the structure from the X-ray diffraction pattern. So oxcarbazepine succinic acid crystallizes in a monoclinic space group with the single unit cell consisting of 8 molecules of oxcarbazepine from 4 molecules of succinic acid and 4 molecules of the chloroform was used as a solvent here. This is the crystal structure, single crystal structure, and this is the layered structure of the 3-dimensional. The 3-dimensional structure of the oxcarbazepine and succinic acid, we can see how the hydrogen bonding are there between the drug and the co-form. We can see the hydrogen bonding between the drug and we can clearly see the hydrogen bonding. So this is the dissolution profile of the multi-component solid forms of fluorocarbazepine. Now the next example is of course there are but one more. Lenalidomide is an anti-cancer drug used in the treatment of multiple myeloma. So we have prepared the co-crystals. Lenalidomide has pH-dependent solubility with low solubility at higher pH. So it has poor permeability also. This motivated to explore the potential of lenalidomide to co-crystallize with an aim to enhance its solubility and permeability. profile and turn its oral absorption. So prepared with linolenamide with vanillin, linolenamide with malic acid, linolenamide with 2,4-dihydroxybenzoic acid was prepared by solvent assisted mechanochemical grinding method. So these are the anyhow, this is the DSC scan of the co-crystals. These are the hot strain microscope. So these are the FTIR spectra, and there are some changes in the major in the frequency of the functional group which are responsible for the hydrogen bonding. PXRD pattern, again there are some appearance of some unique peaks. There are appearance of the unique peaks in the co-crystal shows that these are the new phase has been formed, and these are the crystal structure. This is the three-dimensional crystal structure prepared by this. So we can see that there is a hydrogen bonding between the drug and the co-former. This is for the 2-hydroxybenzoic acid and the drug and the malic acid and malic acid, and then drug and the vanillin. You can see the hydrogen bonding that in the co-crystals, both the drug and the co-former, they are attached to the hydrogen bond. These are the prepared, and they all have, and these pictures we have taken from the X-ray detectors. So in the case resolution profile of the co-crystals, solubility and permeability enhancement, and then we have done the pharmacokinetics and the cell inhibition responses of linolenamide and co-crystals. So we have done the effect of co-crystals on the cell line, and there is an increase in the effectiveness of the co-crystals of the drug in the co-crystals as compared to the pure drug. Now, do we have time? Should I move further or should I stop here? Please.
Yes, ma'am.
No, please continue.
Continue, ma'am.
Yeah, one thing I want to tell you that, as I have told you earlier, that we have our intention to prepare a co-crystal, but sometimes instead of having a co-crystal, we get either a eutectic or we get a co-former, and the same thing has happened within our lab also. So actually, we wanted to prepare the co-crystals of a drug, and instead we get the other form. The combination of multiple therapeutic agents into unit dose has become a popular drug development strategy for the management of many complex disorders such as infectious disease, HIV, cancer, diabetes, and cardiovascular diseases. So fixed dose combination products can comprise simple drug-drug combination for the effective treatment. So we also wanted to combine two drugs to form their co-crystal. We wanted to combine two drugs instead of co-former and a drug. We wanted to combine two drugs so that we can have a co-crystal of the drug-drug co-crystal. So what happened? We get one co-crystal, one coamorphous, and one eutectic. So our focus was to develop some drug-drug co-crystals comprising two or more theoretically effective components in a stoichiometric ratio within the same crystal lattice, wherein the components may interact via non-ionic interaction, preferably hydrogen bonding. So this is a traditional approach. This is the multi-component solution. Therefore, what you do, work was initiated with an aim to prepare drug-drug co-crystal of three poorly soluble anti-hypertensive drugs, telmisartan, irbesartan, and hydrochlorothiazide. Now, keeping the objective in mind, atenolol has been selected as potential co-formula because it belongs to the beta blocker class that plays important role in treatment of cardiovascular diseases and the hypertension. So we wanted to combine it with the atenolol. Atenolol possesses three amide groups that have high propensity to form hydrogen bonding with selected drugs based on complementary functional groups. So what we did is that these are our selected drugs. Atenolol was taken as a reagent and we have the atenolol, hydrochlorothiazide, irbesartan and telmisartan. But what happened, you can see the atenolol with telmisartan, we could prepare a drug-drug co-crystal. And with hydrochlorothiazide, we get only the drug-drug eutectic. If we don't characterize it properly, so one would say that, it's also a co-crystal. But no, it was not a co-crystal, it was a eutectic. And what about with the irbesartan? We could get a coamorphous. We could get the coamorphous. And we have done it and we have characterized it thoroughly. So we can see that this is the DSC of telmisartan atenolol co-crystal. Telmisartan, and this is the co-crystal. This is the co-crystal DSC, and then coamorphous atenolol and irbesartan, and this is no peak corresponds to the melting point in the DSC. That shows that it is a amorphous, and then when we recrystallize it, even in the DSC, we again heated it and recrystallized in the DSC cell only, and we get the peak. Then after recrystallization, after recrystallization, it has come back to because we know that amorphous form is always very unstable form. It will convert to the crystalline form. And it has converted into the pure irbesartan. And now it is the thermal stability versus of pure amorphous form. So coamorphous of irbesartan and atenolol, and it's the pure irbesartan. It is more, we can see that it is the coamorphous is more stable because the pure amorphous irbesartan has been converted into the irbesartan crystalline form. But in the same condition, our coamorphous was still very, very stable. It means that it can be used for the formulation. It is stable as compared to the pure amorphous irbesartan. So DSC of the hydrochlorothiazide and atenolol eutectic. You can see this is the hydrochlorothiazide, atenolol, and this is the eutectic. The melting point of it in most of the one thing we must know that if in the case of eutectic, the melting point of the eutectic is always less than the pure drug and the co-formula. Whereas in the case of co-crystal, it may be in between, or it may be a bit, in some cases it is lower. But in the case of eutectic, it has to be, it is always lower than the co-formula and the co-crystal. And in the case of salt, it is always higher than the co-formula and the co-crystal. So these are the few things we must notice when we are preparing our multi-component form. So this is the phase diagram of our atenolol and hydrochlorothiazide eutectic, the phase diagram. It was prepared with the help of DEC only. So now, when we saw the FTIR, first we could compare it only. We could see the DSC in the coamorphous form. We get only the no peak was there, and in the case of amorphous. Technically, the melting point was below the drug and the coke and both the drugs. And in the case of co-crystal, the melting point was in between the drug and atenolol and both the drugs. But you can see the FTIR of telmisartan atenolol co-crystal. We can see that telmisartan shows stretching vibration of carbonyl free carboxylic acid at 1697. Atenolol shows bonding vibration band of NH3-amide group at 1648. Now the ground product of telmisartan and atenolol show characteristic sharp band at 1673 that is different from the both. It means there is some interaction between the two. There is some interaction between the two. And in the case of co-amorphous, shifting in NH bonding and asymmetric NH stretching of tetrazole rings of irbesartan from 1616 to 1667 and 3368 to 33, because some short range interaction for amorphous may be some short range interactions are there, of course not long range, but short range interactions are there. There is some changes. And in the case of hydroxide, there is no changes. There is no changes in the frequencies, any of the frequencies. Now, even in the case of PXRD, shifting of position in the prominent peak from 6.7 to 7.9 in ground product indicates the formation of new crystalline phase in the case of telmisartan, atenolol co-crystal. Absence of characteristic peak of atenolol at 9.9 shows that a co-crystal has been formed. Now, this is the crystalline pattern. This is the pattern of co-amorphous, and these are the crystalline of the irbesartan and atenolol. Irbesartan and atenolol, and this is our co-amorphous. This is the now in the case of eutectic, we have the peak. We have the no change, no shift in the peak, no appearance of the new peak. But eutectic is just a mixture of both the peaks. Because there is no interaction. There is no interaction in the eutectic. So we have the peaks of both the components, atenolol as well as hydroxide. They are intact in the case of PXRD. So this is the crystal structure of atenolol and telmisartan, because I have told you that a structure has been formed. This is the so homomeric interaction among molecules of telmisartan. This is the homomeric interaction in the telmisartan, homomeric in the telmisartan molecule itself. Homomeric interaction among molecules at atenolol, amide-amide dimer, and now when the co-crystal is formed, the homomeric interaction have been broken and heteromeric interaction has been formed. Heteromeric interaction between telmisartan and atenolol, NHO hydrogen bonding shows that a co-crystal has been formed. You can see that these homomeric interactions have been broken and heteromeric interaction has been formed, and this has been done with the single axial diffraction. So these are the postulated heteromeric. Postulated, not we are, but we have postulated that because some short range interaction may be there. So we have postulated that postulated heterosynthesis between irbesartan and atenolol. These are the this is the three-dimensional hydrochloride and atenolol molecules. This is the structure in the weak interaction between hydrochloride and strong cohesive interaction leads to eutectic, because there is a strong cohesive interaction between the hydrochloride and atenolol, and here the heteromeric interaction was weak, So therefore, both the homomeric interactions are intact, and we get the eutectic, and we get the primary sulfonamide catamers change in structure of hydrochlorothiazide, presence of atenolol molecule with the free amide complementary group could not able to interact robust sulfonamide catamer chain of hydrochlorothiazide to build any new supramolecular network, and instead of getting a co-crystal, we get a eutectic only. So the summary of the various multi-component forms were co-crystals, types of solid, and the nature of interaction: additive, heterosynthesis, homomorphous, additive, nature of interaction: additive, heterosynthesis form, postulated. This we have really seen in the crystal structure. Here it is postulated. Now in the case of eutectic, only the homosynthesis are there. Homomeric interactions are there. Amide and primary sulfonamide catamers are there. So this is the aqueous solubility and improvement in solubility in the case of coke 82-fold. In the coamorphous, of course, it is 42-fold, and eutectic is 13-fold. But why we are go for the co-crystal? Because the co-crystal are stable and crystallized. Amorphous form, everybody knows that they are unstable, and eutectic are also not very stable. So this is the physicochemical parameters, the ideas and improvement in solubility, and maximum decrease in blood pressure was observed in coamorphous system compared to other system due to its higher solubility. Percentage decrease in the blood pressure we have determined. So in conclusion, I will say that the crystal engineering approach was successfully applied to development of multi-component solid form as well as multi-drug solid forms. Last decade has witnessed the extensively exploration of this field for mitigating the pharmaceutical issues such as solubility, stability, related bioavailability, and permeability. Besides, the use of computational tools further boost in the designing and development of multi-component solid forms. So computational tools are that we can see the structural database. We can see the Cambridge structural database, and now we are using the Cambridge structural database to see whether there is a possibility of forming a hydrogen bonding or not. Okay, so these are the list of patents we have filed. Some have been granted also for the co-crystal of metanine, co-crystal of naringenin. We have genistein, and it is, I think, naringenin has also been granted. Then these are all the patents I have filed, and some of them I have got also. This is my student. He has done with the atenolol, and he has prepared all the three eutectic, one eutectic, one coamorphous, and the co-crystal. And all my students, he is in the Dr. Reddy's lab. He has had biotech. All my students, they are all well settled. Some of the old students. These are my new students. They are working.
Thank you, madam.
Yes.
Thank you, ma'am, for providing an insight of a development of co-crystal using crystal engineering approach, and also the possible alternative outcome of co-crystallization, that is eutectic mixture. Thank you, ma'am. Now, ma'am, we have certain questions that is asked by our delegates. Can I recite, ma'am?
Yes, please.
Now, the question is asked by Yes, the question is that how can we differentiate eutectic and co-crystal? By which method, ma'am?
I told you the eutectic, and the first method is that we must see the melting point with the help of DSP. In the case of co-crystal, in the case of eutectic, the melting point will always be below the drug and the co-former. But in the case of co-crystal, it may be in between, or it should be in between. But sometimes the melting point may be below both the drug and the co-former. In that case, we should see the physical mixture. In the case of physics, if we take the physical mixture, we don't grind it, kuch na kare hum, we just mix it and see the DSP. If it is a eutectic, we will get a single peak. But if it is a co-crystal, it is going to be a co-crystal. In the case of physical mix, we will get two peaks. Two peaks, one to the co-crystal, another one is the drug, another is the co-former. In the case of if it is a co-crystal forming, then the physical mixture will give us two peaks, not a single peak. This is the one case we must do. And then we should do the FTIR. In the case of co-crystal, there is some changes in the major functional groups which are responsible for the formation of hydrogen bonding in the co-crystal. But as in the case of eutectic, there is no hydrogen bonding, the FTIR will be intact. It will be the same as that of the co-former and the drug. There will be no changes in the FTIR. There will be no changes in the powder X-ray diffraction also. It will be the same. So therefore, whenever we have a eutectic, we must first of all see the melting point with the help of DSP, not on the simple melting point approach. It's with the proper DSP.
Thank you, ma'am. Now the second question that is asked by a faculty, that is Rajeshri madam. How can eutectic be differentiated with those co-crystal having a lower melting endotherm compared to their pure form?
Yeah, I'm telling you. The eutectic, it's by nature, eutectic has a melting point lower than both. But in the case of co-crystal also, the melting point is low. So then we have the confusion. In that case, I have told you that we must take and we have been publishing the data also like this, that you take and then referees will tell us that how do you say that it is a co-crystal. Then we'll say that then we give the physical mixture. In the case, if it's a eutectic, the physical, suppose our drug melts at 240 and our co-former melts at 140. Then if we take the physical mixture, if it is a co-crystal, then we will get the two peaks in the DSP, but it is not at the same position, it will be at a lower position. 240 की जगह पे हमारा आ जाएगा 230 या 220 आ जाएगा, और वहां पर 120 या तो हमारा 118 आ जाएगा, दोनों का कम आएगा, ठीक है. But if it is a eutectic, we will get, even in the physical mixture, we will get a single peak. In the case of eutectic, even in the physical mixture, we will get a single peak. And then I have told you, you go in for the FTIR, you go in for the PXRD, and there will be no change. There will be no changes in the PXRD pattern. There will be no changes in there will be no additional peaks in the PXRD. No, there is no interaction will be shown. No change, no shift in the vibrational frequencies in the FTIR. Yes.
Thank you, ma'am. The next question that is asked by our faculty, that is Hiral Manani madam. Can eutectics give same kind of spring and parachute effect like co-crystals?
No, but in the case of eutectic, in what happened that in the case of I think the eutectic, they don't give the same spring and parachute effect. We get only in the co-crystal, because in the case of co-crystal, the co-crystal and our co-crystal, our drug is attached to the co-former, and they both together they will go to the solution. They will go to the solution. So we have not noticed the same spring and parachute effect in the case of it. It is only in the properties of the co-crystal.
Thank you, madam. Thank you for answering all the questions, ma'am. Now we are towards the end of our e-seminar. Thank you, ma'am. Now, the end of a story is a new beginning. Yes, sir.
Thank you, madam.
Okay.
Okay, the end of a story is a new beginning for many more. So on this note, let's conclude a remarkable, memorable, and knowledgeable events. For concluding remarks, I would like to invite Ms. Hiral Manani, Assistant Professor and Coordinator of this event. Over to you, ma'am.
Thank you, Krishna madam. Good afternoon to one and all. I, Hiral Mannani, coordinator of seminar, going to give you brief summary of today's session. In first technical session, Dr. Kelvinson has given very good view on importance of multi-component system and classification and advantages of co-crystals. Also sharing the advantages of co-crystal and binary phase diagram of the co-crystal. In second session, Dr. CVS Subramaniam has enlightened depth knowledge on value addition strategies of pharmaceutical product expansion, utility, and bioequivalence requirement, also about the limitations and advantages of new therapeutic application. In third technical session, Dr. A.K. Bansal explained very important topic during seminar about co-crystal as a novel solid form for formulation development, comparative overview of co-crystal with other solid forms, case study of co-crystal, base product also describe the evolution of pre-formulation properties and oral bioavailability with study design. In last technical session, we have gained depth knowledge from Dr. Renu Chadda on development of multi-component solid forms using crystal engineering, characterization, design of co-crystal, and crystal structure analysis. Also about the combination of multiple therapeutic agents into the unit dosage form. Thanks. Now, I would like to give vote of thanks to all the eminent dignitaries who have spent their valuable time to make this event successful. First, I express my sincere gratitude to GUJCOST for financial support and organizing this one-day international conference. It's an honor to have Dr. Bhupendra Singh Chudasma, our Honorable Education Minister, Government of Gujarat, to grace the occasion with his presence, and we thankful him for sharing his thought and motivation. I also grateful to our chief guest, Professor Dr. Naveen Seth, Honorable Vice Chancellor GTU, for his kind acceptance to grace the event as a chief guest and for his inaugural words. Thank you, sir. I am also extremely grateful to our beloved Honorable Vice Chancellor, Saurashtra University, Dr. Nitinbhai Pethani, for presiding the occasion. Despite of his heavy schedule, he has given kind enough to accept our invitation. Thank you, sir. My thanks also goes to Dr. Vijaybai Desani, Honorable Pro Vice Chancellor, Saurashtra University, for his presence, inspiration, and support at the event. Thank you so much, sir. My thanks also go to Dr. Jatin Soni, Registrar Sri, Saurashtra University, for his presence and grace the occasion. Thank you so much, sir. Now, we have been fortunate to have of the renowned identity from academic in different areas of subject, Dr. Kelvin sir, Dr. CVS Subramaniam, Dr. A.K. Bansal, and Dr. Renu Chadda for sharing their valuable thoughts and knowledge with us. Thank you so much, all these eminent speakers. Building up of an institution of this size in indeed a Herculean task that has been continuously accomplished by Dr. Mihir Rawal. convener and head Department of Pharmaceutical Science, Saurashtra University. I heartily thank to you, sir, for constant inspiration and support. Thank you so much, sir. I also thanks to Dr. Sachin Parmar, Department of Pharmaceutical Sciences, Saurashtra University, the co-convener of this event, deserve a big hand of applause for overall supervision. Thank you so much, sir. I also extend my sincere thanks to organizing members, Dr. Nazir Varia, Dr. Krishna Karodia, Ms. Priya Patel, Ms. Mosmi Bagela, and Ms. Jinal Adhya for their support to make this event very successful. Moreover, I would like to thank my colleagues and all the participants from different institutes of the country to make this event successful by their valuable presence. Lastly, I would like to thank our computer center team for providing continuous support and timely help throughout the event. Thanks a lot to all members. Once again, thank you all and have a safe and healthy life. Thank you. Thank you, all of you.
Thank you very much to all and all the speakers of this seminar for making this event very much informative and successful. Thank you very much.
Thank you all. And have a nice day.